Sir, you can start now. We are live on YouTube.
A very good morning to everyone and a very warm welcome. It is indeed a great pleasure to welcome you all to this event titled Genetical Decoding Life, with a pun on the word genetical. This event was originally planned for 2020 as an in-person event. Unfortunately, as the COVID-19 pandemic gripped the entire globe in the early months of last year, our hopes of hosting this event were shaken. However, I'm really grateful to the Royal Society of Biology UK, who has sponsored this event, for being so supportive and allowing us to transition to this online mode. And luckily, the Indian National Young Academy of Sciences has been a wonderful platform and has been organizing such programs for like past so many weeks and months. So we have already gained the confidence to go ahead with the support of INYAS, especially the Kolkata Bhubaneswar chapter. INYAS is the first and only recognized young academy of sciences in India and was founded as the junior wing of the Indian National Science Academy Council in December 2014, with a vision to promote science education and networking amongst young scientists at the national as well as the international level. Such an event to motivate young minds towards science is in perfect alignment to the objectives which INYAS stands for. And the Indian Institute of Technology Kharagpur was also very happy to support this initiative for genetical decoding life. And so now here we are. Just to brief you about the event, this is basically aimed towards school students, especially from class 8 to 10th, and with a focus on the basics of genetics and how these concepts are changing the paradigm of modern day healthcare. The event aims to motivate participants towards understanding the role of genetics in health and disease, and will include interactions with experts in the field. This will also be followed by a quiz competition on topics related to high school genetics. So here we have 6 wonderful schools participating in this program, and they include Aditya Birla Public School Bhubaneswar, DAV Model School Kharagpur, Don Bosco School Kharagpur, Griffins International School Kharagpur, Kendriya Vidyalaya 1 Kalaikunda, Kendriya Vidyalaya 2 Kalaikunda. The program includes 2 wonderful interactive sessions with our professional experts. And so we have Professor Praful Shukla, Assistant Professor, School of Medical Science and Technology, IIT Kharagpur, who will be discussing about the basics. And then we have Dr. Rashmi Sinha, consultant gynecologist and obstetrician, who will be talking us about the applications of genetics. I encourage the students to put on their thinking caps when listening to our experts and make it more interactive. So anyone who wishes to ask a question during the session may please click the raise hand button on Zoom, and they can unmute themselves only when they have been asked to do so. So that will help us keep it more submissive, and it will be easy for us to coordinate the entire event. And now let me move on to introduce our first expert, Professor Praful Chandra Shukla. So Dr. Shukla did his schooling from Central School and then his bachelor's in science in chemistry and biology. After that, he paid more attention to modern day biology and hence chose master's in biotechnology, thinking that it is the future of modern science. But according to him, he was partially correct and partially wrong. It is not the biotechnology, but an overall understanding of biology that is needed when you study basic subjects as well as medicine, particularly medical, clinical, and biological sciences together. So then he chose for cardiovascular diseases topic for his PhD and learned a great deal about research in modern medicine. He did his postdoc from Toronto and Boston. Subsequently, he joined BITS Pilani and then IIT Kharagpur. Presently, he's working on some aspects of the cardiovascular system, specifically heart and vascular diseases, and how genes affect the disease of heart and vessels. He loves to teach and encourage to do experiments. He has received the American Heart Association and American College of Cardiology plaques for being the finalists in the Young Investigator competitions. And so without further ado, I would hand it over to Dr. Shukla to please go ahead and start his presentation. Dr. Shukla, please.
Thank you, Dr. Chakravarti. It's wonderful to see the students here today. And before I start, I will just share my screen. Let's see. So please let me know when you see the screen.
Yes, it is visible.
Okay. Yeah, so good morning to all of you.
Good morning, sir.
Good morning. I welcome all of you to this lecture on decoding life, and more specifically the genetics. Today we are going to see that in the biology, what we have in the store, and how we understand biology. Specifically, when we talk about or when we hear the term biology, the next moment we think that we are talking about plants, animals, viruses, bacteria, and the long names that we have to mug up in this subject, which is not actually true. And sometimes looking at the traditional way of studying biology, we are a little bit disappointed and put off because of the lack of interest, because in the biology, unlike mathematics where you have 1 + 1 = 2, we don't see that happening in the biology. So today's lecture is focusing on how biology is, how interesting subject it is, and sometimes it becomes a little bit challenging to understand the intricacies of the biology that we, modern day biology, I would say. So I'm sure that some of you might have seen this movie as well, in which a person's genetics is changed by the scientist, and now the person has got immense power and strength to do wonderful work. And many of you, when you see these kind of movies, you try to think or you try to imagine that probably someday you may also be able to change ourselves and our genes in a particular way. I also thought so, but I was incorrect. So coming back to biology, there are a lot of students who will choose mathematics, who will choose physics and chemistry. A lot of students also choose biology, but to me, it's the combination of all the subjects that you study, and it's a pinnacle of what you see around us is because of all those fundamental sciences coming together. So when we see the things or the term here, the biology, it's like plants, bacteria, virus, animals, and also humans. I will also request you to remember this slide, and we will come to the end, and I will ask you to remember this slide and then connect all the dots together. So one experience that I have from my school days is that people were little bit apprehensive about going to the biology because they had very long names. And to clarify that thing, I would like to mention here that if we cannot identify what it is, we cannot establish the relationship between the things. For example, if I tell you that I'm saying 100, so anywhere in the world, if you go, if you say 100, people understand. You will understand that 100 means 100, nothing else. But in biology or in this kind of sciences, if you have not identified it properly, say I'm putting here a picture of peacock. So when we say in India peacock, we can clearly imagine what the peacock is. But if you go to South America, probably they will not know what peacock you're talking about. So it is very crucial, it is very essential to identify and properly document and name this item, thing, or whatever it is in this type of kind of subject, so that you can clearly establish a relationship, which is also very important for us to understand the history or the natural history of species or living organisms. So this is about as a subject, but we are talking today about the genes and genetics, and this is one of my very dear subject. So many of you have heard the name of Mendel, right? I am sure that you know the Gregor Johann Mendel, which is also called the father of genetics. He was a priest in Austria, and he was interested in questions I just mentioned to you. That he selected what was available at that time. He wanted to know what is the relationship between these living organisms, how these living organisms, whether it is plant or an animal, how they are related, why they are so different, why one flower is pink and one flower is white. So these kind of questions, when you start thinking, when you try to answer these questions, you will start looking for the ways to answer these questions. So this happened also the same thing with the Mendel. He was a priest, and in the backyard of the church, he had this plantation, and he started to look for what is the reason why there are so different plants, why there are so different pea pods, because these were the pea plants that he was having in the backyard. So he carefully studied the phenotype. I will just tell you what is phenotype. Phenotype is the external features that you try to, or that you identify the organism with. That means when we are talking about phenotype, I can tell that Gregor Johann Mendel is having 2 hands, 10 fingers in the hand, dark hair, this much height. So these are phenotypic features. And when we talk about genotype, then we are actually talking about what is inside, what are the genes inside the organism. So he started to look into the phenotype, and then because he was very curious about these features and how you can control, so he started to cross these pea plants together. So you see in the picture that he's taking a brush and a knife, and then he's actually cutting the part of the flower that is important for auto-pollination, self-pollination. So if you don't allow self-pollination, what will happen? You can control the pollination with the other types of flowers, and then you try to find or to see the result, what may happen, what kind of plant this will be developed into. So after lots of experiments, like probably number of years, he came up with the 3 laws of inheritance. These laws are, the first law is law of segregation. The second law of Mendel is law of independent assortment. And the third is law of dominance. So I will tell you very briefly, very quickly, what these laws are. So law of segregation says that a gene or an allele, for that instance. So before going into what is allele, so I will just tell you what alleles mean. Alleles are the alternate form of a gene. So I will come to that point later when we are talking about genes. So alleles are different forms of one gene that we have received from our parents. That means in humans, we have 2 alleles for every gene, one coming from mother and another coming from father. In the first law of Mendel's inheritance or law of segregation, that it says that these genes or these alleles are segregated when their gamete formation is. So at the time of this when the time of gamete formation in the humans, these genes are randomly segregated. And when they are fertilized, then they come together. So, I will tell you with an example in the next slide. So this is the law of segregation. You are taking here, for example, this flower has red color, and this is denoted by RR. So here the capital R is written. Capital R means this is the dominant phenotype. And if I write a small r, whenever you see small r, that is the recessive phenotype. So these phenotypes, the dominant phenotype means if there is only one allele R, this will be expressed. So now you can question, what is expressed? I don't know how many of you know what this expression means. Expression means, when gene is there, it results into some kind of features to the organisms. It gives a certain kind of features to the organisms. So the gene R, gene for the color, is having two alleles, either R or small r, and then it can also have RR, capital RR, or a small rr. So in this case, the capital R is generally in biology, in genetics, is denoted as dominant gene. And when this is small r, for any phenotype that you can decide, small is denoted as the recessive phenotype. So in this case, one R is sufficient to carry out the function, and it gives you red color. When you have a small r, that means it is not able to give you the red color. That's why small r is denoted as the recessive phenotype. So what law of segregation says, that during the time of gamete formation, these R and R are separated, segregated. Segregation means separation. So these R and R are separated, and when they combine together in fertilization, based on what kind of genotype is, you will see the result of the color, result in the form of color. So in this case, R and R, where is the capital R is the dominant phenotype, this will be expressed in the color red. The second law of independent assortment is very interesting law actually. So this tells you that there are two features, the genes or alleles for two different features are not going together. They are independently going to the gametes. What do I mean with this? Is that, for example, you are a very tall guy. It may not, and you are also a very intelligent person. So if I assume that the height is based on one gene and your intelligence also is based on one gene, then these genes in your kids will not be going together. That means your kid might be tall, or your kid might be also very intelligent. So this law of assortment says that these two features may not go together if they are not located very close to each other. If you have any doubt, you please raise your hand, because this is very interesting to understand and very important as well. The two features which are not, the genes for the two features which are not located close to each other will be independently going to the organism, to the gamete, to the offsprings. This is law of independent assortment. And the third law is called law of dominance. That means, when you have a dominant gene present, that will give you the characteristic rather than recessive gene. That means you're not going to get the effect of a small a, because if the capital A is giving you the red color to the cherries, whereas a small a, if both of the small a are present, it will give you not the red color. So this is by default, if you have one a, it will give you the red color. So that is law of dominance. That means if you are having a gene say for example, if you are having two alleles coming from your parents, that is for your, if I assume that this is, your hair color is based on only one gene, and if you are getting B for capital B, say capital B for black color of your hair, and small b is for not the black color of your hair, it can be anything else. So if you are getting both from your mom and from your dad, if you are getting BB, or if you are getting from your mom and from father, it is B, you will be getting only black color of hair. If you are getting B from your mother and B from your father, you will not have the black color. So this is law of dominance, that one gene, which is the dominant gene, gives the main characteristic to the organism. So I also told you about the alleles. So this picture tells you that alleles are different forms of the different versions of the genes that code for a trait, a phenotype, a characteristic or a feature of the organism. So here in this case, you can see that the color of the flower, which is the phenotype, is being given by different alleles of the same gene, which are imparting that different colors to the different combinations of these alleles when they are expressed. What Mendel did was he chose pea plant, and he was lucky in that case. He had the pea plantation in the backyard of the church, and he started to look for different features of the different parts of the pea plant. One is that this is the seed, right? So the seed is either round, which is a dominant phenotype, or a wrinkled, that means having some folds on the cover of the seed. So that he gave the term wrinkled for this. Then you have seed color, which is either yellow in color when they are ripe, or they are green in color. Their flowers are also different. The pea pod shape is also different. You might have seen in your house in the winters when you get the pea pods for food, you might see the differences in the pea pods. Some pea pods are very small, very tightly packed, and they are constricted. And some pea pods are very big, though they are having small seeds inside and they are very smooth. So these kind of things you can actually, if you can control the pollination with the different cross-pollination with the different types of plants, you can control these features. And you can identify what will be the result. You can actually predict what could be the result. So this is what he did in 1865.
So Professor Shukla, sorry to interrupt. We have a couple of questions here. So
Okay.
The first question is from Saksham. He's asking, he's not clear about alleles. And then the next question is from Shona. He's asking, sir, can you explain the location of allelic pair in a chromosome?
Okay, so alleles are, let's take the simple example of us as humans. So you have, so I'll just draw it here. So you have a chromosome, a pair of chromosome for each. So one chromosome you are getting from father, one chromosome you are getting from your mother. Alleles means, for example, there is a gene which is present here. The same gene, another copy of this gene will be present here. These are called alleles. What may happen, what happens also, these genes which are present, these alleles which are present for the same gene may have slight differences among them. May have slight differences in terms of their sequence, in terms of their expressivity. That means, one can be highly expressed or slightly more expressed than the other. That is what gives us the differences. We are not completely equal to our father or mother because we are getting the mixed results of the alleles. So alleles are these. So in the position, if you are asking for the position where they are located on the homologous chromosomes. Because we are getting one chromosome from father, one chromosome from mother, and we have 23 pairs like this. So in each pair of the chromosome, at the same location, these alleles are present. Is that answering your question?
Thank you, Professor Shukla. I think that's quite comprehensive.
Okay.
And we have some more questions, but we'll take some of these relevant questions towards the end of your talk.
Okay. Okay. So this was in 1865. He identified, like he published a small paper, which was not, that much got the recognition at that time. So moving forward, coming later, 80 years after this, what Mendel found out, at the time of Mendel, there were other people who were also trying to identify the reason behind differences in the organisms, but they were probably not so lucky and they did not choose pea plant, because what the pea plant was probably a very smart choice he had, or it was just a chance that he got the pea plants and he got the very nice results. Other people could not get that, and they were, we know some of them are there, but not so much recognition they got. So 80 years later, people came to know that yes, it is the DNA. So at that time, nobody knew about the DNA, and it took really long time. So for the first time, when the DNA was identified, it was named. The name was given as nuclein. So this nuclein was isolated from the bandages of the patients, and he isolated something which is acidic in form. And Frederick Mischer, I think he gave the name nuclein. 80 years later, I can say, about 150 years later of that case, Griffith, with a very smart experiment, demonstrated in 1928 that yes, it is there is something which transforms one bacteria. If I can move this one, there's something changes from one bacteria to another bacteria, which might change the phenotype. So let's see for this example. So you have a strain of S. pneumoniae. So this is a bacteria. So this is the bacteria which he took, and when he injected this S strain, that means a smooth strain of this bacteria into the mouse died. He chose another strain which is called R, that is rough. When he injected this strain of the bacteria, mouse did not die. What he did then, he heat killed the S strain. He heated the S strain by heating. We can assume that these things will be killed. And when he injected this heat killed virulent strain, that means which was causing the death, it did not kill the mouse when the bacteria strain was heated. That means you kill all the bacteria, there's no more there. But when he mixed the live non-death causing bacteria with the heat killed S strain, and when he injected, the mouse died. See what you can infer from this experiment. This is very clear. It is very clear that something is being transferred from this one to this one, which is causing this non-death causing bacteria, non-virulent strain, into a virulent strain. And that he gave the name as the transforming principle. We still don't know that what is it. At that time it was a little bit of confusion that what it could be. So some years later, different groups, they started to identify what is it, and they came up with the final conclusion that it is DNA, nothing else. The transforming principle that was transforming one non-virulent strain of bacteria to a virulent strain was the DNA that was causing the effect. So in 1944 and 1952, during that time, there were several groups which were able to demonstrate and conclude that yes, it is the DNA. How did they do? There were several experiments. One experiment I'm showing you here, that from the S strain, from the virulent strain, they isolated all the components separately. They isolated RNA, they isolated protein, they isolated DNA, and other components of the bacterial cell. and then they injected or they used this separated components of from that is S strain. They try to transform the R strain, which was a not dangerous bacteria, to the mouse. So when they separately use these components, only the DNA was able to cause the transformation of the R strain to an S strain, and then it led to death of the mouse. So this was a very confirmatory experiment in which they showed that is the DNA which is causing the transformation of the bacteria. So now you can imagine it took about 150 years from the first time when the nuclein was identified and when the DNA was actually known to say, or it was confirmed that this is the genetic material which codes for different features of the organisms. So before going a little bit more into the details, I would just like to here tell you about the levels of organization, because this is also very important to understand. Different levels of organization and different division of task or labor among the cell or among the organisms. So this slide tells you that there's a cell. When the cells form, the cells come together, they form a tissue, and then tissue is different types of tissue form an organ, and then organ system, and then the whole body, like that. So whether it is plant cell or an animal cell, you have this type of organization visible at all levels. So if you talk about cell only, this organization, so this example I'm telling you, so this type of organization that you see, I'm just giving you an example here for your skin. So if you zoom in of your skin layer, you will find different layers of in the finger, for example. If you go deeper, if you zoom in more, you will see different types of cells which are lying at the level, at that layer of the skin. If you zoom in more, you will see different types of different cells are closely or tightly packed together to make this layer, which is very important for us to be protected from the outer attacks. This is the skin layer that keeps us safe from the pathogens that are living around. If you zoom in more, you will see different organelles. So this is called mitochondria. I think all of you know. So this is mitochondria. If you go deeper, you will see that this mitochondria, as well as in the cytosol, you have a lot of these ribosomes are there. I will come to that point where I will tell you exactly what these functions are in a couple of minutes. So this is what a cell looks.
Just one more time, Professor Shukla, I think there is one student who has raised the hand. So
Okay.
Can we just ask him? Shiva?
Please.
Yes, sir. Good morning, sir.
Good morning.
So my doubt is that in genetics, so why can't genetic tests predict all the diseases?
Why can't the genetic test predict all the diseases?
Yes, sir. And sir, I have one more doubt, but I'll ask it later.
So this question, you will get answers automatically for this one. Let me go for 2-3 more slides and you understand why.
Okay, sir.
Is that okay? What is the next question?
Yes, sir, it will work. So my next question is that in the term, the identical twins, it's given that it's identical, but still, sir, they don't look the same. Why so?
Right. So that also you will get answers.
Okay, sir.
Yeah. So I knew that you will have these kind of questions to me. So I was telling about the organization of the at the very small level to a very high level. So this I'm telling you about this is a picture of cell in which in the center you can see this is the nucleus, and inside the nucleus you have a lot of DNA, and this nucleus is surrounded by the reticulum, endoplasmic reticulum, and then this is mitochondria, this is Golgi. So this is a structural organization which is you will find in nearly all the cells like that. That in the center there's a nucleus, that is the governing core of the cell, where all the codes are located. These codes are being read by the system, by the machinery. Then they come out of these pores. These are nuclear pores. These they will come out from these pores and will be expressed. Again, this expression term we use a lot in biology or in genetics. Expression, please understand, means that it has been the information has been transferred in the form of code to a form of protein. So that is called expression. So these whatever they come out of it and they are expressed, and that is what decides the phenotype of the cell, the function of the cell. For example, if you are talking about liver's function is very different from the function of the blood cell, that means RBC. So liver produces a lot of enzymes. So if, for example, if this is the liver cell, this will be producing a lot of enzymes, a lot of proteins, a lot of other things for our detoxification, for metabolism. Whereas our blood cell, having the same DNA in the beginning, will not be doing these functions. They will be just carrying the oxygen to and fro. So based on what genes are expressed, the function is determined, and that is another level of organization, the systematic organization in the living organisms. So what is this DNA made up of? DNA is made, I'm sure that you might have seen CSI Miami, or probably some other fiction movies in which you may have heard the term DNA, or as well as in your classroom or books. So DNA is actually a polymer made up of primarily these base and then sugar and a phosphate group. And the picture that you see here is that this is a ladder shaped molecule in which these both strands are running anti-parallel to each other. That means the polarity, we denote it with this 3' to 5' and 5' to 3'. This is because the location of the OH group that is on this strand. So these strands run anti-parallel to each other, and if you twist this ladder very much, then you will get the DNA, what it looks like inside the cell. And if you zoom into this ladder, you have this, these are bases. So this is a kind of nucleotide that is present. One nucleotide is attached with the phosphodiester bond in a polar manner, in a polarity. This gives you a polarity, and how this is organized gives rise to a DNA. So DNA is deoxyribonucleic acid, and these are the bases that are located. So there are only 4 bases. So imagine our genome has about 3 billion base pairs. So what is base pair? Base pair means you see this anti-parallel strands of DNA are running together in opposite direction, and they pair with each other. So here there is another pair that will be attached to this one with the hydrogen bonds, and pairing happens between A and C, or A and T, and G and C. So there's a double bond between adenine and thymine, and there's a triple bond, hydrogen bond, between guanine and cytosine. And this is the whole code book for the DNA, for our genome. So our genome contains 3 billion base pairs. So now if I answer your question, what you asked, we have 3 billion base pairs. How you are going to identify the changes in the DNA or the sequences with these many base pairs for every cell? I will also give you more clear answers in subsequent slides. So, and this DNA is actually, linked. So these every base is linked with the phosphodiester bond that is provided by the either with the 5th carbon here or 3rd carbon here. So that's why this polarity is for the DNA. And this picture tells you that, this A pairs with T and G pairs with C. That's the whole thing about the DNA. One interesting fact I would just like to tell you, maybe it gives you a sense of scale. So if you take 10 bases like that, so 10 base pairs, so this is called base pair. If you take 10 base pairs like that, like this, so the length is about 3.4 nanometers. So all of you are very good in mathematics, I'm sure. So you can tell me at the end. So this 10 base pairs are the size, the length of 10 base pairs is about 3.4 nanometers. And we have 3 into 10 to the power 9 base pairs. That means 3 billion base pairs in one cell. So just calculate how much would be in one cell if I keep end to end all the DNA molecule from one cell only, how big, how long this DNA will be. You just calculate it. And one more interesting fact is that we have about 30 trillion cells in human body, about 30 trillion cells. So now if I take the DNA out of each cell and keep end to end, how long that would be? You please calculate. So I can tell you, this will be in meters. So it is like 3 4 12 and 3 3 9 and 1. Say about 10 meters. So length of DNA from one cell will be 10 meters and you cannot see the cell. And you cannot see the cell, but this DNA is packed into this cell. See how it is packed. It is very interesting to see, and it is all very physiological for the cell as well to pack it in a such manner that it is not being available for it to express all the time, and it is being governed by the very complex mechanism that everything is not amenable, not achievable to be expressed. So I just gave you here example that how this long DNA is expressed. So we have 23 pairs of DNA, right? Chromosomes. So each one chromosome is one single polymer of DNA. One chromosome is one molecule, and that is one molecule is being packed in such a manner that it can be compacted in the form of a chromosome. I can tell you later in subsequent slides. This is very interesting and very different. There's a different branch of biology or genetics that deals with this kind of system. So what is genetic code? So if I talk about, if I just told you that
again, sorry to unmute.
Yes.
So there is another person who is raising hand, Mashraba. So maybe a relevant question is this, so I just wanted her to.
Yes, please.
Yeah. Mashraba, if you can please unmute.
Yes, sir. Good morning, sir.
Good morning.
Sir, actually, my question is related to the law of dominance. Sir, basically, sir, is a male gene more dominant or a female gene more dominant?
It has nothing to do with this.
So, like like a male gene can be a dominant or a female gene, both.
Yes.
So then, why we generally hear in our society that when a daughter is born, she inherits more from her father, and when a boy is born, he inherits more from her mother.
So this is if you talk about the biological aspect of this, so if you have, I just told you that we have 23 pairs of chromosomes, and so I'm not telling you that about the philosophical reason about what the society thinks, but in the terms of biology, we have 23 pairs of chromosomes. Out of these 23, 2 are sex chromosomes, right? So you have, if you have 46 chromosomes, you have 44 plus 2 sex chromosomes. So these 2 sex chromosomes are very different. They are termed as X and Y. So when just if you look carefully, everybody either will have, so this is combination for a male, and XX combination will be for the female, right? So I told you that they are independent. So these genes are going independently when the gamete is formed. So when the father is forming gamete, it will form X, one gamete, and another gamete carrying Y. And mother is going to form only X because both of the alleles or both chromosomes are X. So it will give you X and X only. So when this X and X combines, gives you daughter, and when this X and Y combines, this gives you what? A male. That is the biology behind the. So I don't know what you at the last statement was that father is. What did you say? Father is giving rise to more characters to the girl?
No, sir.
Yes.
Sir, actually, we generally hear when a female child is born, like we hear from our family members that she will inherit more from her fathers, and if a boy is born, he will inherit more from her mother, like facial features.
No. That is not true. That is not true. That is completely random.
So that can be considered as a societal myth or something like that.
Yes. It is completely societal myth. It is never, you cannot generalize this one that it will happen. It's just a chance event. And because we are getting genes from both of them, which one is expressed during the time of development when the embryo is developed inside the body, at that time, which set is expressed more, those things will be, those features will be dominating in the person. It does not have to do anything with the law of dominance.
Okay, sir, understood.
Okay.
Yes, sir.
Okay. So I was talking about the genetic code. So genetic code, I'm not going into codons here. I'm just would like to tell you that this, if you take this all the genes or all the sequence of the cell from our body, from one cell, from humans, you will have hundreds of books can be just filled with the sequence. So imagine the size of the DNA or amount of DNA that we get, amount of code we carry in each cell. So I think Shivam, you asked me a question that why cannot we do genetic testing for each disease? First is that diseases are not entirely caused by the genes all the time. There are genetic diseases which can be due to the defective genes, but then it's completely based on what is available, what is feasible, and whether you want to know it or not.
Yes, sir.
Right. So we have so much of information, and everything cannot be deciphered or determined for everyone. But this happens if you ask me question, why cannot we do genetic testing? That people in whose family there's a disease running for generations, they undergo genetic testing to know whether they also carry the risk or not. If you're not having any family history, it is just a waste of money that you will be going for, right? And you may not know at the end, you will have different genes. So what your dad has got, you might have slightly different genes. Your dad might have some disease, but you might not have it. I'm not telling you that you will not have it, you might not have it. So then it is very difficult for you to determine whether the gene that you have received from your parents is causing you or going to cause you some disease or not.
Yes,
It is very difficult to say. for all the diseases though.
Okay, sir.
Okay.
Thank you, sir.
Yeah, welcome. So, it is, I'm just telling you, I'm just giving you a sense of the enormity of our genetic code in each cell and how these things are managed by the nature throughout generations, throughout the evolutionary history, that we have managed to, successfully carry out over the, millions of years, that, genes are being progressed and they are still evolving, but there are many more conservations. That the conservation is also that continues to, have our genes, get our genes safely transmitted from one generation to another generation. And it is the nature's, miracle that we get if we have only one mutation. For example, if I tell you that, sickle cell anemia, this is one example that we always get. Sickle cell anemia is caused by mutating. So you have billions of genes. Not everything is coding for something, but they also are just lying there. There are different reasons for it. But there are genes, thousands and thousands of bases long, and only one change from C to T or A to G will cause mutations in the protein that will give you the disease. That may happen. But this doesn't happen all the time. You see how effectively, how nicely system has arranged or organized in a way that it transfers everything very successfully without any big problems. So going forward, I will just tell you that we have genes. These genes are in the form of, this is actually DNA, and this DNA, when it has to be read, so we get the codes from the parents or from the cell in the form of DNA only, and that is being translated into the protein, which is the workhorses. That means whatever we are, whatever the way we look like is because of the proteins that we are getting from the expression of this DNA. So this is called central dogma. That means DNA gives rise to RNA, and RNA is giving rise to protein. Right? So this is the central dogma for all the living organisms that happens, nearly all the living organisms that are, that we see around, in humans, in dogs, in cats, everywhere. And this is how so a set of, say, I told you an example of liver. So in liver, if you are thinking that there are like, I'm just giving you a random number, say 15 genes are in the form of DNA are expressing, maybe in neurons, that will be another 15 genes. So not the same genes will be expressing. It will be different set of genes expressing and give rise to a nature. So these two are very different system. This is the function of, this gives rise to the function of, detoxification, metabolic activity of the body, whereas these neurons are for your activity, the brain activity, or your movements, which are governed by the brain. So these are governed by or conducted by the neurons. So these different set of genes are expressed in different type of cell from the same DNA. So we are having same DNA in all the cells, but which gene is expressed at what time tells you or determines the function of the cell. So for example, also, if somebody gets a black eye or a blue eye or a brown eye, is also depending upon what gene is expressed, what gene it has received from the parents, and what set of genes are expressed, that will tell us the phenotype. This is the phenotype. Whether the person is having, connected ears or free earlobes, or person is having blue eyes, or they can roll their tongue, or their black hair or brown hair. All these things are based on what genes are expressed. There you can think of the dominating genes, whether it's a gene is dominant or gene is recessive. Same holds true for the diseases as well. The diseases which are based on the genes. So I'm just telling you some examples here of the proteins that we know. So proteins are also acting as enzymes, right? That we have like a lot of enzymes in our body in each cell, and these enzymes perform different functions. Even in the plants, although we have enzymes, there are structural proteins that gives the structure or a shape to the cell. That means whether it's a hair or a skin or eye lens or nails, whatever these are, or heart, so they give structure to the cell. So these are proteins. Transport proteins, the proteins which carry different types of cargo to and fro within the body. It can be hemoglobin, it can be cholesterol laden with the like protein mixed with cholesterol, also transport things from one part of body to another. Motor proteins, say by which you run, which you work, able to work, your heart is working, you can walk. These are motor proteins, and there are motor proteins inside the cell which carry one metabolic or metabolite from one part of the cell within the cell to another part of the cell. That are also motor proteins. Storage proteins. We also have storage proteins in our own body. Basically, these are not storage proteins that we generally get in our body, because this is one thing that in human body we cannot store proteins as such. You have to keep on building it. Otherwise, that you cannot store in the form of protein in human body. But there are other places where you can these can be stored in the form of protein. Say for example, milk. So milk has a protein, or eggs. They have proteins which we used to eat, but they are not meant for eating though. But we used to eat to get more protein, because our own body cannot store proteins as such. Our body can store glucose very nicely, fat very nicely, but not the protein. That's why you have to keep on eating proteins all the time. And there are signal proteins. So these are the proteins which organize or manage the signals between and to the cells. So signals we are getting from one part of the body to another one gland to another one. These are being carried out by the help of proteins. So proteins are everywhere. That is what makes us. Receptor proteins, how we see light, whether we see light and darkness or we see color, is because of the proteins. This is happening. Gene regulatory network, special purpose protein. There are special purpose protein, that means in plants or in some animals there are proteins which makes them safe. For example, in plants, when there's these proteins are expressed, it is protected by the very cold temperature. Same as in the other sea animals, when they are in a very low temperature, they are still alive because these proteins are helping them. So little bit forward, so we know that we are getting genes from our parents, and you are sitting in the class. Maybe all of you are very, very similar in terms of, if I look at the genotype, that means the sequence that you have got from your parents are almost similar, 99.9% similar. Whereas if you talk about chimpanzee, they are like 96% similar to the humans. Imagine the similarity between these and how different we look like, how different we behave. What is the difference in intelligence between us? That is just the 4% of the genes, or 4% of the proteins that we are getting from those genes is making us so different. A cat and a human is 90% similar. And so as even banana is 60% similar to humans. But see the differences in the phenotype. So this is, this is the miracle of the nature, or the biological kingdom that has evolved over the years, that it has, by tweaking, turning on and turning off the expression of the genes at a point, it will just give you a very different phenotype. What genes? So far I've been talking about genes and genes and genes. What are genes actually? The genes are located in the chromosome say this example I'm telling you, that this is one chromosome or one strand of, long strand of DNA, and that will have several genes located at different places, which are spaced by a part of the DNA as well. So this is gene A, for example, this is gene B, gene C, which are very, very different in the sequence, and their size, and even in their functions as well. So gene A, according to the central dogma, will make RNA and will make protein A. And in a cell, if you can imagine that now the complexity comes, first is the sequence is different, second is that their function is different, and third, how much they are expressed in one cell. If this is in one cell expressed 100 copies, and this is 10 copies, this is 5 copies, makes you a liver cell. For example, in another cell, it will be like 5 copies of this one, 25 copies of this one, and 9,000 copies of this one will make another type of cell. This may happen. And this happens all the time. So based on this, you can imagine the complexity of each cell. We have got, different types of cells in our body performing different types of functions. This is just because of what genes they express. They all got same chromosome. They all got same DNA, but they are not expressed similarly. That's why we have very different cells in the body. So this is this example is the yeast genome, which is very small part of the DNA from the yeast, and you see how many genes are there in such a small part. How these are packed? So I just give you a very nice analogy. You might have seen people go to the parlor to get their hair curls. So this is exactly the DNA is packed. You have the DNA, and this yellow part is called histone. And this DNA is, so you have double strand DNA. This will be wrapped around this histone, and then these histones along with the DNA are wrapped in a very closed manner. Then it will be more compacted, and then you got the chromosome. So imagine the length, you have 2 nanometers here, and you have 1400 nanometers here, the 1.6 micron, 1.4 micron, this is the thickness, approximate thickness of the chromosome. So how compactly this has been and efficiently this is being packed. Imagine what will happen during the cell division, where these chromosomes are separated and they go to each daughter cell very efficiently. This is electron microscope of the same thing, how these, so this one is called nucleosome. That means the DNA wrapped around histone is called nucleosome, and these are nucleosomes that you can see here in the electron microscope, and they are packed more in a very compacted manner. And this is another picture. So moving fast forward, we are coming to the 21st century. How much time we have left?
I think we are
a couple of more minutes.
No, that's fine. Please take your time.
So very fast forward, as I said that we are 99.9% identical. If you are sitting in the class, you may be your genes are so similar that a thought sometimes comes: can we clone the humans? So can we clone ourselves so that we don't have to, even if I die, there's a clone of me which can still live. So this has been the thought. This has been the science fiction movies. You might have seen that people clone each other, and then they live, or they may have different features and powers, things like that, which is not absolutely wrong, absolutely incorrect, but also ethically it will be disastrous to clone humans. Yes, sometimes human cloning has been done. I wouldn't say it is a cloning. So actually you have to understand that cloning means 2 different types, or 2 different meanings of clonings are there. One is that you clone like a person is cloned in exactly in the same way, right? So you clone it like SIM card cloning. So people sometimes make a copy of the SIM card and they clone it. So this is one cloning. A second cloning is that you take a part of DNA and you put inside a vector. So this is also a kind of cloning. And this is generally we use every day in the lab, not this one. So we don't make copies of organisms by cloning because this is a very different thing. Whereas the cloning that we do in the lab is that we take a part of DNA from, say, mouse, and we clone it into a vector, which is also called plasmid. So I'm not going to details of this one, but these are two different types of clonings we come around. And so in the human cloning, people who cannot, who have problems in producing babies because of some reasons, because of some disease or because of other complications, they sometimes go for cloning. And this is also not called cloning, this is called in vitro fertilization. What happens is that they take the donor egg, they remove the nucleus. So in the in vitro fertilization, this doesn't happen. So you take the donor egg and then you fuse it with the another donor for a sperm, and then you put into the surrogate mother. Surrogate mother is the mother which is not giving to either to egg or the sperm, but it is just holding the embryo inside. So that is called surrogate mother. So that is in vitro fertilization, but this type of cloning is possible. People have done in the dolly, in the frog, that they have taken the egg from the donor, they have removed the nucleus. Now this is ready to receive another nucleus from an adult, and after this is being combined, this is now the egg having the nucleus from another donor, that is giving to the DNA, the 2N DNA. Normally what happens is the egg have N DNA, and your say egg and then the sperm is also having N DNA. But this cell has 2N, which is complete in itself. So when you replace this nucleus which is having N chromosome, if you replace the 2N chromosome, this is a complete embryo that you can put into the surrogate mother. It will be developed into a baby. This is not allowed ethically in many countries, but there are some countries which it is allowed only for 6 days. So you cannot go beyond 6 days of development. So that is of no use, but this is possible. Theoretically, it is possible. People have done this in dolly as well as frogs species. And now I think they have done in the monkeys or in the cats as well. Another use of this is very interesting, that the animals that have got extinct, if you get the intact DNA from the cell of those extinct animals, you can choose another similar phenotype or similar genotype or similar animal and place into the surrogate mother where this extinct animal can be revived. So this is being tried for several extinct animals. There is some success, but not 100%. So this is my personal opinion. It is better to have robust physical and mental health than clones or longer life. I will just conclude my thought. I will just say that it is, rather than doing cloning or having our copies so that they can live longer, is not correct, but having good health which comes from genes and our choices and the environment where we live, and that is what gives us the health. Why I am telling you this is as an indication of that we are actually a living, walking universe for the bacteria or other species inside us that makes us a complete human. This picture tells you that we have about only 30 trillion human cells. The rest of them, about 100 trillion cells, are something not human. but we harbor them. So you can now say that we are not actually us completely, but we are a mix of genes that we are getting from our parents, as well as whatever is living inside or on us that makes us the full, complete human. Why I am telling you? People have now identified the fact that there's something called microbiome. This microbiome is so important that without that, we may not even function normally, the way we function, the way you see to function. And without them, we cannot actually probably going to live if they are not there. So this is what makes us healthy, complete human. And this is very important to understand that, the microbiome that we harbor inside our body or on the body is very important for a lot of our things. Nowadays, people have identified that they can affect the diseases, they can affect our behavior, they can affect obesity, and a lot of other things. Whether you believe in one political party or other political party may also be related to the microbiome that we have. One example I will just tell you, and then I'm finishing my talk. So in 2019, they identified that people who run long distances, they harbor a specific bacteria in their gut, which makes them, or which allows them, or which enables them to run longer. What they did, they took that bacteria, put into the mouse, and mouse was also able to run longer. Similarly, people who are living longer, that means who are slowly aging or aging very healthily, they are also found to have a special type of bacteria inside, which makes them live longer without any complication, without any diseases. So this is what I want to convey. I think my last slide, that this is very important to eat healthy so that you have a very healthy microbiome, because this microbiome affects our genetics as well. How genes are expressed and how they will be affecting our phenotype is not completely human, but it is a mix of human as well as the microbiome that we have. With this, I will try to would like to close you, and thank you for your attention. Thank you very much.
Thank you so much, Professor Shukla, for that wonderful talk. And you have, I think, covered most of the things which are of interest to young participants like we have.
I hope I could.
It was amazing. We have quite a few questions, as I can see on the chat box right away, and we did pick a few questions. I would like to take a few more from here on. So first, I would like to go ahead with Ajay Tripaঠি's question, and this is quite interesting. So, why do we have these recessive? Why do these recessive traits exist, and what is their application in an organism?
So these recessive traits, the question should not be why, because it is just we are evolving every day, and during the selection process, during the selection of the genes which are favorable to us, they are still selected. The dominant, so it is not that it may be that something which is dominant in one population can be recessive in another. So you cannot say that these are why they're existing. It may be different for the other population, for other people as well. I'll just give you one example, that the blue color of the eye is the recessive feature, whereas the black is the dominant feature. Now you go to the places where there is less sunlight. That's how their genes have been modified in such a way that they express those which are helping them to live at that area of the earth. Whereas for us, it is probably it's better to have darker skin so that we can avoid getting too much of exposure. So these are actually selected over the time, and they are not why they are existing. There has to be there is a reason for them, because they give selective advantages for organism to survive or to live normally. One example I can also give you that many times say there was one cross country this Olympic winner who used to ski and won, I think, 4 or 5 gold medal. He had a mutation in one of the genes that makes blood cells, erythropoietin. So this guy had a mutation in the erythropoietin gene, which was making more blood cells, 20% more blood cells over there, without any artificial or external causes. His gene was mutated, but this gene is not found in others. So you can say that this is recessive and it is not present, but for others it might be beneficial because it is going to help you if you are going to go for cross-country skiing or high on mountain, high altitude running. Then you need more oxygen. So then you need more blood. So by this mutation, you might get more RBCs so that it is enabling you to run longer, which is not found in others. So these are kind of chance event, and then after the chance event, they are selected over time to give added benefits to the organism. So the question should not be why.
Thank you. I'll pick up some more questions and maybe collect a few questions together and ask you. That will be probably helpful. Okay, just a moment. So one question is from Shyam Maiti. He's asking, can there be more than 2 alleles of a single gene? And then another question is from Shonok Mazumdar. He's asking, can humans be a pure breed after several attempts of breeding? So if you could just discuss these, and then maybe I'll take some.
Okay, so the first one is that yes, we can have several different alleles for the same phenotype. Say ABO blood group, right? So we have ABO blood group. So there are 6 alleles for this. So this is possible. Many times we see in the plant that there are several alleles for same trait. But in humans, normally we get 2 alleles for one gene. But if you look at this example of blood group, whether the person is getting A blood group or B or O, it has got 6 alleles to decide that. Right? What was the second question?
Yeah, I'll just go back to it. So somebody is asking, can humans be a pure breed after several attempts of breeding?
So I'm not going into the humans, but I will tell you about the mouse. Yes, in the mouse, what do you mean by pure breed? That means, first tell me what is pure breed. What do you mean by pure breed? Who has asked this question?
So in that case, I would request the student directly to please go ahead. Please unmute yourself. I think it was Saksham, right?
Saksham, can you unmute yourself?
I'm sorry. I think it is. Let me just get back.
No, sir, I had not asked this question.
Okay. It doesn't matter. Shonok Mazumdar. Yeah. So I think if he's not able to unmute.
So I will tell you. I mean, pure breed is something very metaphorical, I would say. But in the mouse, what people do to rule out any variability among the species, they have been crossed from last 100 years within themselves. And with this breeding model, they have come up with the mouse, which is, if you take 2 mice from that breed or 10 mice from that breed, they are genetically same.
Yeah, I think that addresses the question a lot. Let me go back to the other questions. So in the interest of the time, probably we'll have to restrict a few questions. I hope that is okay. Yeah.
So Deepti Goswami is asking the paired alleles of a gene separate randomly during gamete formation. So I'm not sure if that is a question. So Deepti, if you can unmute yourself and
Yes,
please
sir.
let.
Actually, I was asking, sir, good morning, sir. Sir, I
Good
was
morning.
asking to just give a brief explanation on of the law of segregation.
So law of segregation is that, because I told you that the two alleles are located, for the same genes are located on two different chromosomes, which are homologous in nature. So you have chromosome 1 coming from parent, one like father, and one coming from mother. So chromosome number 1. So you have the two different forms. So the gene is same, but there might be some sequence change in that gene. That's why they are called allele. So they are located in two different chromosomes. When the gametes are formed, then one chromosome is pulled out and the other chromosome is pulled out on a two separate daughter cells, and these are going to be future gametes, future egg or sperm. So that is how these two are separated, and this is what exactly is the law of segregation is.
Thank you, Dr. Shukla. Let me go back to the questions. So I think there are a few questions which might be relatively more relevant to our second speaker.
Okay.
We'll move on to, we'll skip those, but I think there was one question which I saw. Just give me a moment. Okay, yeah. So Shyam Maiti is asking that in the law of dominance, one allele is more dominant than the other. So how there should be two chances? So, there should be two chances, tall and dwarf. But many people are what we call normal, average height, or yeah. So there is a range of heights that we see. How is this possible?
So I that's why I added a term, assuming that one trait is being controlled by one gene, right? So you are taking an example which is of height, which is a multigenic trait. When you are the height is being decided not only by one gene but number of genes together, right? So even though you have segregated and you have got for some, so height is actually a not a very nice example for dominance, because it is being controlled by several genes at a time. If you talk about, say, blood group, ABO, then you can say yes, if the person is having a particular dominant gene, that blood group will be there, because this is completely dependent on a particular allele or dominant allele or the recessive allele. But the height is multigenic. So in genetics, we study a lot of multigenic traits, multigenic orders. Right. So this is this is not a very nice example, not appropriate example actually for the height. That's why we see variations in.
Thank you, Professor Shukla. Yeah, I do see there are some more questions, but I think I at this stage, like non-coding part of the DNA and all they've asked. So I think this may not be the most appropriate platform at this stage. So we'll skip those questions because they are more of an advanced discussion that is needed. So that's definitely.
So I actually didn't know that they will be having questions on non-coding and non-coding genes.
So if you would like to discuss, you can just go ahead.
If they have any questions, let me know, please.
So it's just they're asking regarding what is this non-coding part of the DNA. So maybe just a 1 minute discussion on that.
Yeah, very briefly I can tell you that so we have if you take 100% of our genome, only less than 2% is coding for genes. I showed you one example of the Earth and how DNA, if you take end-to-end DNA, how much it would be. So it will reach from one part of Africa to another part, but only hardly is very small part, all the genes will be there. So our 97.5% of genome does not code for the proteins, and which is not coding for the proteins called a non-coding genome. Though it does not code for anything, but it do affect a lot of cellular processes and function and the phenotype. Now we are slowly started to understand that these non-coding part which are expressed, they don't code for protein, but they take part in regulatory processes. And that's why they are very important.
Okay, thank you. Thanks a lot, Professor Shukla. It was a very exciting session that we had, and I'm sure the students and the teachers and all of us thoroughly enjoyed the session. It was
Thank you very
amazing, and
much.
I'm sure there are questions flowing in, and there are hundreds of questions in the minds of the students. So we probably will have to organize a separate session as well, so we can discuss on that. Thank you so much.
Thank you very much.
So
Thank you.
with this, thank you. So with this, we move on to our next speaker now, after that exciting session by Professor Shukla. So now we have Dr. Rashmi Sinha. So Dr. Sinha has done her MBBS from Patna Medical College and her master's in surgery in obstetrics and gynecology from RG Kaur Medical College, Kolkata. She has also got a diploma in minimal invasive surgery from Germany, and she's a renowned gynecologist in Kharagpur. So besides her private practice, she's presently a visiting consultant at 4 Airports Hospital, Kalaikunda Airfield, and Bharatiya Reserve Bank Node Mudra Private Limited Plant Hospital, Shaboni. Previously, she was an assistant professor in the Department of Gynecology and Obstetrics at Dr. B.R. Ambedkar Medical College in Bangalore. Besides clinical practice, Dr. Sinha has been, she has a very keen interest in research and teaching as well. And she likes to motivate young children towards science and education. She truly believes that the path to our success as a nation is only through education. And so now, without further ado, I would like to hand over the floor to Dr. Sinha. Dr. Sinha.
Yeah. Thank you, Dr. Chakravarti. Hi, good morning, everyone. First, I would like to thank the organizers for organizing such a beautiful platform for the students, for the young minds, and especially the team genetical decoding life, and the organizing team, especially the Royal Society of Biology, UK, in INYAS, and IIT Kharagpur for organizing such a wonderful initiative to train our young mind towards genetics. So last 1.5 years was the year of COVID. So during COVID time, we have understood what exactly this genetic engineering can benefit or can harm in many ways. So now Dr. Shukla has already beautifully explained you about, it was a very explicit explanation about the genes and the genetics and the genetic engineering, what exactly the research is going on towards this field. So so now I think you must have a brief idea about what exactly the genetics mean, or the genetic engineering, and what the progresses are already we have achieved in this field. So the main idea of this program is to introduce you with the success and achievements that we have achieved in this field, and to take you to a magical world where the engineering is being performed on the living things, the living organism. So first, I'll share my screen to share the slides with you. So let me know if the screen is visible to you. Yes. Is it visible to all of you?
It's not yet visible. Just please try to share the screen.
Okay.
So on
Just give me some time.
towards the lower part of, yeah, you have to share the entire screen.
Okay. Okay. Sure. So now it's visible?
Yes. Now it is visible.
Okay. So first of all, I'll just make it full screen. Okay, now first of all, I'll that the genetic engineering as such, that success that we have achieved, most of these success are in the field of agriculture and fishery. So here we have achieved a huge success. So the list is very long, if you can say. So I'll just introduce you with few of them. So this picture is, you might have seen this picture. If this picture is basically showing golden rice. What exactly these golden rice is? This golden rice is basically a genetically modified rice to enrich our nutrition with vitamin A. This is basically vitamin A. What actually this vitamin A does, I think most of you know it's very essential for your growth and for your eyes. So this rice is been modified to enrich your grains, enrich your rice with the ample amount of vitamin A, so that you don't get deficit of these vitamins. So now, do you know actually what this aquaculture means? If anybody of you have an idea about what this aquaculture means, so if you don't know, it's okay. It's aquaculture actually means the culture of the aquatic organism. Aquatic means the organism which lives in water. So actually, what happens in aquatic organisms, we have modified many of the fishes. So one of them, these fishes are being modified for many to play different roles. Those different roles that is for the increased consumption, for the productions, for the they are being genetically modified to as a sentinel for the pollution, that is a marker for the pollution, and they are also being modified to decorate your aquarium. So one of this example of this tadpole is what you can see on the screen is a tadpole. Which tadpole is, it's not the normal tadpole. This tadpole is genetically modified tadpole, and from where the gene has been taken, this gene has been taken from jellyfish. So jellyfish, what exactly this jellyfish basically flourish. So the genes from this jellyfish, they had taken, our researchers has taken the genes from this jellyfish, and they have transmitted this gene into the genetic sequence, genetic codon of this organism, the tadpole. So what happens once this tadpole is a special tadpole, this tadpole basically detects the level of pollution inside the water. So what happens once pollutants means, you know it better than me, that pollutants means that some biochemicals, some minerals which is harmful for us and for the organism inside the water also. So what happens when these chemicals, these tadpoles are being exposed to these chemicals, then it causes a level of stress inside their body, and out of this stress they start flourishing. So this way we can get to know that what the level of pollution is there inside the water, and to take action according to that. This slide is basically showing just the thing that how we have increased the production for the consumption of the food, consumption of the fish. So if you don't have idea, we have already these fish. One of this fish, I can say that its name is aqua salmon. So aqua salmon fish is basically available in the market, and US FDA, or you can say it's some regulatory body is there. They have already allowed for the consumption of and the production of these fishes, and they are already in our market for the consumption purpose. So this way, what happens in these fishes, these are the small fish, and these fish are being genetically modified to convert it into the fishes that can grow faster and into bigger size. So this way we are increasing our production towards this way. Again, the slide is showing that I think you have marked for last, I think in last 20 years, we have seen that our aquarium are being beautifully decorated with some flourishing fishes. So what are they exactly? From where did they come? They are basically again the genetically modified one, and they are being modified again with the same jellyfish to flourish. And now they are looking so beautiful with the yellow, red, and red, pink, red fish like that. So these are again the modification on the genetic base, on the gene base. Now, this picture, it doesn't seem so beautiful after this, what I had shown you, the beautiful aquarium look of those flourishing fishes. I think you might have seen these pictures before also. So what are they? They are chicken. They are not disease. They are basically genetically modified chicken, and they are featherless. So why did these scientists have modified these fishes, this chicken? This is for their increased production, again for the consumption purpose. So what the benefit they are getting after doing this modification? These chicken can grow better in warm climate. In warm countries, they doesn't need any cooling, and so this can increase their availability and their production in the warm countries also. And now you have heard a lot of buzz about the pollution everywhere, the pollution of the land, water, and pollution in the air. So one of this component of this is water pollution. And water pollution, you can say it's an inevitable consequences of our economic growth. So what happens usually? The industries and the factories, they are dumping their waste in the water, in the sea or the river, and along with that some oil spills, because oil spills happen sometimes in our water. So because of this, what happens during these oils forms a layer above the level of the water. So what happens there? The oxygen that the organism are getting, they are not able to. So because of that, their survival is very difficult. So these are, as I said, it's inevitable, but our scientists are doing a lot. Our scientists are tirelessly working on it to combat the solution for this. So in this field, again, they have made some genetically modified bacteria. A few of them are Alcanovorex or Marinovorex. These bacteria, what does they do? They basically break down these complex hydrocarbons so that they can clear up the level of the oil layer above the water, and so that the organism can get a beautiful environment, and then they can get oxygen for their survival. So if you are not aware of this, in this field, which I would like to mention it here, have you heard the name of Dr. Anand Chakravarti? And he had done the patent was, he had the first person to get the patent in this field. I mean, the patent on the living organism, it was not been possible before, but he is able to get the patent towards the living organism. The living organism is a bacteria. The bacteria is the Pseudomonas, and this bacteria is again again, it works on the same pattern, like it clears the water pollution and by breaking down these complex hydrocarbons. The list is so long, so it's not possible to explain each and every like aspect of what the modification was being done. But I would just like to enumerate a few more of this in the field of agriculture, like this pest-resistant cabbage. What these actually actually this is basically a cabbage which is being genetically modified with the scorpion poison, so that they the caterpillars cannot attack. Once they will attack, they will get the poison of the scorpion, but this scorpion poison is not at all harmful to us, to human. This is again the genetically modified eucalyptus tree, which is been modified for the to tolerate the freezing temperature of the many countries, especially in Australia. You can get this eucalyptus trees. After this agriculture and fishery, we have now we will come to the true clinics, and this I believe that this will be much interesting for you. So in clinics, what exactly we do? What is the use of the genetic engineering? So genetic engineering, we can say it's you can understand this in two ways, like two aspects of it is there. So with genetic engineering, we are increasing, one aspect is that we are increasing the production of some drugs, some hormones, and some vaccines. So this is the one aspect. It's all about the production. So production to cure diseases, for the to improve diseases or to cure diseases. And the second aspect is to actual gene therapy. If you have heard this word gene therapy, it's the true therapy we are in. I'll tell you in detail after this. So it's the true gene therapy that we are introducing some genes into a human body or a human cell. So first we'll come to drugs and vaccines. So among these, you have heard a lot about, I can say in last 1 year, you have heard many things about the these drugs like the remdesivir, molnupiravir, and favipiravir. So what exactly these drugs are? So these drugs, I think from the social media, you can you were better aware of these drugs. The remdesivir was that was a huge threat that they are not getting this drug because it's the production was not sufficient at that time. So these drugs, what do? They actually they work on the genes, not exactly on the genes. They will modify the genetic they will modify the production from the genes that the proteins and all those components that are being produced by the viruses. They will just attack on it. They will check the production of those proteins from the viruses, so the disease can get cured. And Zolgensma, if you have heard, I think these days for last 4 or 5 months, we are, if you watch television, you are seeing many celebrities are raising the funds for the patients suffering from the spinal muscle atrophy. So these, what this spinal muscle atrophy actually is. This disease is basically a disease where those babies are just born. At that time, they have a sufficient amount of these SMA proteins, but that is sufficient for that small babies. But once they start growing, these proteins are not sufficient for their growth. So what happens usually, this what this SMA protein does, this is the protein to maintain the tone of the muscles. So what happens with this, the proteins are not there, so they are not able to maintain the tone of their body, so they are not able to walk, they are not able to speak, they are not able to, and at last what happens, once this protein get deficient in the respiratory muscles, so you can better understand that what happens if the respiratory muscles are not working, they are not able to breathe, and as a result they won't be able to survive for long. So what this Zolgensma does, so this is again the same way, it increases the production of this SMA protein 1. So with this SMA1 protein, they have a better chance of survival. I cannot say that this is the complete cure, though the companies are claiming, but they can prolong the life of the baby. Now, coming on to after drugs, in the production part, I had told you the drugs, the vaccines, and hormones. So drugs I have explained, and now the vaccines. The vaccines, it's like you all know about the vaccine. In the childhood also, you had taken so many vaccines to protect yourself from the various diseases, and in the last 1.5 years, you have seen many vaccines are in the market. Like Covaxin, Covishield, Pfizer, and lot many more. So what does they do? They work on the same similar to they work on the mechanism of the genetic programming. Like what happens with the vaccines, vaccines are of various types. So few of them are true viruses, mean the attenuated short of, means they are not active, but they carry the viral various proteins and the virus itself. Like you have heard that the Pfizer is the mRNA for spike protein, and this Covaxin is the whole virus that is attenuated. So in this way, we are challenging our body with the vaccines to produce the antibody against it, and so that we are able to combat with these diseases. Now coming to the production of hormones. If you are not knowing many of them, I believe that you know one of it, like insulin. Insulin is, I think everybody knows insulin, because many of in your relatives or in your family, somebody might be suffering from diabetes, and you must have heard that they are taking insulin. So what exactly this insulin is? This insulin is again the product of the genetic engineering. So genetic engineering, how does it happen with this? So in the year, I think this was the first hormone to be synthesized in factories, or you can say in the laboratories. So what happens in the industries, it was in 1978, as much as I know, in 1978, it was industry has started the production of this genetically modified insulin. So recombinant insulin. So what happens? They had taken the insulin gene from the human, and they transfected the bacteria with these genes, and then the bacteria, they started producing the production of the insulin gene, and as a consequence, insulin from the insulin genes, the protein started going the proteins, and proteins from there we extracted the hormone, that is insulin. So many more are there in the hormone. Like we in our field, we use many growth hormones, will increase like the in infertility treatment, we use many hormones like the FSH, LH, and all these are what we use are basically genetically modified hormones, because it's not possible to produce these it's not possible to extract hormones from the human body or from the animal. Like, in insulin, once it was like not been produced in the industry, what was been done in those time, it was been extracted, it has been extracted from a pig and pigs and the cows. So in those days, it was what happened, it was very costly and it was not widely available at that time. So this genetic modification or genetic engineering has improved the production and the availability of these genes, so that we can treat many patients at the same time. So for last few minutes, every time we are saying genetic engineering, but what exactly does genetic engineering mean? Genetic engineering is basically a manipulation or alteration in the structure of the gene to create a desired characteristic of the organism or to increase the production. So just a little bit about the history. So if I don't know if you are aware of that word, from where does this term come? Genetic engineering, who coined this term? This term was coined by Jack Williamson. So you, if I'll ask you that, who was he? So anybody have an idea that who was he actually? So you must be thinking that he must be some like a researcher or some geneticist, but actually it's not so. He was a novelist. So he was the writer of the science fiction, and he coined this term in his fiction novel. So from there it comes the word genetic engineering. What now this slide is basically just to show you what actually happens, what we do in genetic engineering. This is the slide which is showing you how we produce insulin in our laboratories. So what happens, the insulin on the left hand side, you can see that the human DNA is there, and from that DNA we are cutting the genes for the insulin. So we are taking those genes for the insulin, and then we are just putting those genes inside the bacteria, DNA of the bacterium. So that is called plasmid. Plasmid is a separate that DNA sequence which is inside the bacteria, and there we are just integrating those genes with the plasmid DNA. So what happens? The bacteria can grow very fast and they can produce a number of types of bacteria, and in this way they can increase the production of genes, and again the genes will translate into the proteins, and from the proteins, from the culture, we will extract the protein, and in this way this insulin will be available to the market. I don't think that this slide is important for you, because it's just saying that what exactly we are doing. In the previous slide we have already discussed. So one aspect of genetic engineering, in the very beginning I had told you that the increased production, and second aspect is, if you remember, it was gene therapy. So production, we have already discussed the production of the drugs, vaccines, and hormones. Now coming to the second aspect of it, it's the gene therapy. I mean, gene therapy, what actually happens here? You might have heard some diseases which is because of the you can say some bad genes, that the genes is not working properly in the body. So how this happens, like those genes are defective, so we will try to correct those genes with the, like, we will take out those defective genes from the human, and then we will correct it, and then we will reinfuse those genes, those cells inside the human body, so that they will get better genes, and then those cells will start function in a normal way. What exactly is needed. So this picture is showing the same fact, like from the bone marrows, we have extracted the cells and we have treated the cells. We have inserted those through vectors, those defective. We have first will cut with the scissors. That is called the, it's not the scissor that we are using, it's the genetic engineering scissors. So we are cutting those things and those defective genes, and then we are inserting the correct genes through vectors inside those genetic makeup, and then we are culturing those cells, and then again it's like infuse those human with the correct cells, and so they will start their function inside their body. In this context, so have you heard about this disease, the bubble boy disease? So bubble baby or bubble boy disease, this is basically the name of what medical terminology. What I can say for this disease is not the bubble baby disease. This baby is famous because this disease get famous with the name of this bubble baby disease because he was the scientist had worked firstly on him for to do some extent of treatment on this baby, but this was not the actual gene therapy what it was done on this baby, but the disease itself is now well known with this baby's name, that is bubble baby disease. And what is exactly this bubble is? Bubble is basically because what happens in this disease, severe combined immune deficiency, it's a disease. What happens in this disease that our body is not able to make antibodies or any cells that can prevent us from the various viruses or bacteria or any other bad exposure. So this baby was treated with some immunoglobulins at that time, but it's not exactly the gene therapy. So I had just shown you this picture so that you will get to know that what actually the disease was and what commonly this disease is being called. So this gene therapy for this SID was this SID means the severe combined immune deficiency. This was the first disease on which there was a successful gene therapy was conducted. So it was in year 1990 when this has happened. So first gene therapy, though it was successful, but it had prolonged the life of the baby, but not the long-term survival. So there are many after that, like after 1990s, there are so many gene therapies are like successful gene therapies are there. So few of them are they're approved by the FDA also, I mean the organizing bodies also. So it's not that everything, it's just about few diseases I'm just enumerating that you might have heard, and in this field the genetic therapy has already been there, but they are not in the market. The research is going on and there is a good success also in those areas like the thalassemia or the hemophilia. I think you have heard these two. And cancers, yeah, cancers is very common. So you must have heard that the cancer in this field also, the gene therapy there is a good success with the gene therapy. I cannot say that this can gene therapy can treat every cancer. It's not like that. But yeah, the success is there, and the research is going on. I think in future we'll get some cure for the many cancers. In head and neck, I can say head and neck cancer, we have achieved good success, and also in the female cancers like breast cancers. But rest of all, though the research is going on, and clinicians are trying for it also, but but the success is not that good. But hopefully we'll get in few years after, from now onwards, we'll get a cure for many of these cancers. So I think I would like to just, this is my last slide. So I would like to just stop with this slide. This slide is, I'll just convey that this is basically what actually this frog is. This frog is, everybody is able to see this, that this is transparent. And transparent means what? They this is again a genetically modified frog. So what happens that because it is transparent, like in our days, what we were doing, we were dissecting those frogs to see the internal organs, or the how the how the liver, the spleen, or these gonads looked like in those days. But now they have genetically modified these frogs so that you can see without dissecting them, when they're live, and you can see that how the organogenesis, the organs develop, how the liver develops, how the liver, kidney, or these organs, or you can see how the disease develop. Any disease, they can expose it to some bacteria or viruses, or you can see some carcinogens, so can produce cancers, and you can see with your eyes that what happens when the disease develop, how this damages the organs, how it happens, like in the cancer, how the growth happens, how fast it happens, and what exactly it does, how it metastasize, and everything. At last, I would say that genetic engineering as such, it's a boon for us. So, but there are many critics against it. So because they are saying that we are like changing our ecosystem. There is a lot of ethical issues also, and some spiritual issues also. Like they are saying that we are changing the world which the God has made. So beyond this, I can say that those are, it's okay, and they are also a bit right on their part, but what I can see that this, because of these genetic engineering, the research that are going on in this field, because of that we are able to cure a lot of diseases, and we are able to improve the survival of the human. So it's on you to decide what actually the benefits and the harms that the genetic engineering can do. So with this, I'll just finish up, and I'll allow you to ask any questions if you have in this. Okay, thank you.
Thank you so much, Dr. Sinha. I mean, it was amazing to hear about the different developments that have happened so far, and what a lovely note that you have ended your talk on. It definitely leaves a lot of food for thought, I'm sure, for the students. And I can already see that there are quite a few questions here. So I'll probably start with a couple of questions which the students asked the first speaker, and which was more relevant to your part. So Saksham has asked, what is surrogate mother? And another question was, what are some Y-linked diseases? If you could just shed some light. Y chromosome related diseases. Chromosome related.
Chromosome related disease. Okay, so first, surrogate mother. So what happens, surrogacy is like if you know something regarding the genital organs like the uterus or the suppose many of the congenital diseases are there in which the genital organs, means the uterus or the many other tubes or ovaries is not there, or the uterus is not there. In those, suppose if the uterus is not there, so in those cases what happens, they will to get a baby, or to get they will a pregnancy, and suppose a woman has not had uterus, so what do they do to get baby? So they will you can say it's not a proper word, that from the, if they have the ovary, their own ovary, and they have from there we can take those eggs, and then from the partner we can take those male gametes, and they can fuse we can fuse those male gametes and female gametes in the IVF culture, and then they will grow the embryo, and after that this embryo will be allowed to grow in the uterus of the another female, which is not genetically related to them, but the genes that the baby, the embryo is getting from the real parents, means the genetic parents. So in this way it happens, like if the uterus is not inside the mother, they can take the advantage of the surrogate mother, and they can get their their own genetic baby from that woman. So this is actually what happens with the surrogacy.
Thank you, Dr. Sinha. I think we should pick up more relevant questions than the Y chromosome disease for your part. So I would probably skip that question right now. So we do have a few questions. In fact, I'll take the second last question first, which is from Shyam Maiti. He's asking, cancer is caused by the malfunctioning of some genes in our body. The cells reproduce rapidly, so those cells also need to follow the DNA. So can we change the DNA by modifying them to cure a human suffering from cancer? And there's another question, which I will just club to this. Can a disease like Down syndrome be cured when the fetus is inside the mother's womb? If you could pick these up. Thank you.
Yeah, sure. The genetic cancer, regarding cancer, I can say there are many researches going on. Like we have achieved good success in head and neck cancer, in breast cancer, because breast cancer, if you don't know that these are runs in the families. These are not all of them, but some of them runs in the families, and there is some genetic problem like the BRCA1 or BRCA2. These are the name of the genes, so you need not to know like about all these things. So in this field, they have modified those that our researchers have changed, modified those BRCA genes, and then they have put some corrected gene inside those defective genes. So in this way, they have tried to improve the genetic makeup of that person, that female or male, whatever. So in this way, they are trying to treat those cancers by taking out those defective genes and putting corrected genes inside the body. So in this field, yeah, in this field, there is so many they have reached till the phase 3, and hopefully we will the gene therapy will be in the market for the cure itself in few coming years. So with the breast cancer, with the endometrial cancer, ovarian cancer, head and neck cancer, and many more cancer, the research is going on, and the success I think will come with the success in the coming few years. Yeah.
Thank you. Thank you, Dr. Sinha. So I think one of the questions Professor Shukla has already answered, I can see on the chat box. So thanks for that. There is another question which is related to how nanotechnology helps in cancer treatment, which I think probably is a separate topic altogether. So let's not get into that. Uh, so there was one question related to can Down syndrome be cured?
Yeah,
Yeah.
yeah, regarding, I forgot that question. Okay, Downs, exactly, what we do till now, it's we screen for Downs. Mean we can, if the parents are like, mostly it's goes from the mutation from the mother side. So till now, what we do, we screen and we detect and we diagnose those defective cells carrying the extra allele of the for the Downs, that is chromosome 21 extra allele for that. So in this field till now, what we are doing, we are just doing, if suppose if we know some genetic disease that the parents are suffering and they will transmit into their babies, in those cases what we do, that we will select those cells. I mean, we are not able to genetically modify exactly those cells, but we will select those good cells from the through IVF, through what you can say test tube babies in your term. So in during test tube baby, what happens that we can select and we can discard those defective cells, and we can select those good cells, those are not defective in their genetic makeup, and can grow the baby or the embryo from those cells, and can just discard those the defective ones. So many more research are being going on. What exactly you are saying, that to change directly the genetic makeup of the cell. So in this field they are working on a disease. If you don't know, the disease is basically the Huntington's disease, and it's the Huntington's choreum. It's the what happens in this disease, it's the abnormal movements happens with in the babies do some abnormal body movements that they are not able to control themselves, you can say. So in this field the work is going on, and hopefully they will get the trials are going on, so hopefully they will come up with the cure for this disease in coming years. And in hemophilia also it's being done, so not for the Downs till now.
Thank you so much, Dr. Sinha. It has been an excellent talk that we had, and we thoroughly enjoyed it. I'm sure the students have got a glimpse of what the potentials hold in terms of gene therapy, genetic engineering, and we have truly probably served the purpose of this program as yet. So, really, we are very grateful that you shared your thoughts with the students, and questions are still flowing in, but unfortunately in the interest of the time we have to restrict our discussion here itself, and possibly we may move again for some another session sometime later, so that we can include many more students and go ahead with the discussion. But for the moment, thank you so much. Thanks to Dr. Sinha, and thanks to
Thanks a lot.
Professor Shukla as well. uh, so now this is the time to move on to our next segment of the uh, event, which is essentially the more exciting part, I guess, for Dr. Sinha. If you could just please stop your screen share, that will be helpful. Thank you. Um, so now is the quiz competition, I think, for which all the students have been waiting since morning. But uh, fortunately, we had a very great discussion so far. And I'm sure this discussion that you had will also help you in the upcoming quiz. So if you have listened to our experts quite intently, then this is definitely going to help you during the quiz competition. We have a lovely set of volunteers for the quiz competition who will be conducting this as the masters of the event. So without further taking your time, I would request our volunteers, possibly if Shatarupa and Mandrita, if you're here. So somebody may
Yes,
please
sir.
turn on your video and you please initiate the session. Thank you.
Thank you, sir. I'd like to thank sir and ma'am for the interactive session. It was wonderful. And now we'll move to the interesting part of our session, that is the quiz. So I'll just brief you all about the rules of the quiz and who the teams are. So if you guys can also show your videos, it would be better. And I'll just
one more
allot.
thing, Shatrupa, just hold on.
Yes,
Madhavi, because we are starting the quiz session, so if you could, like for the YouTube as well, if you could have the tiled
Yes, sir.
version for the
Yes,
entire.
sir. I have changed the settings. Just turn on the videos means.
Yeah, requesting all the participants to please turn on the videos and please listen intently for the rules that are being told to you, because this is an online platform, so we need to be very careful how we conduct this quiz. So please listen to the rules. Thank you.
Yes, sir. So I'll just allot the team names first. So team A is Aditya Birla Public School, Bhubaneswar. I think you should just take it down with which team you are. Team B is DAV Model School, Kharagpur. Team C is Don Bosco, Kharagpur. Team D is Griffins International School, Kharagpur. Team E is Kendriya Vidyalaya 1, Kalaikunda. And Team F is Kendriya Vidyalaya 2, Kalaikunda. So now I'll just talk about the rules. So each question will carry 1 marks in the first round. So we are going to have 3 rounds of questions. The first one will be basic MCQ questions, and subsequently each round will be an eliminating round. So we start with all 6 of you. Then we subsequently take 5 teams, and for the third or the final round, we take the 3 teams. And also, because it's an online platform and I think 3 members are there from each school, it would be better if each school can give me 1 captain's name, so that in case of any difference of opinion in answers, I take the answer from the captain. So I'll request the schools to kindly give 1 name who I can treat as the captain of that team.
Good morning, ma'am.
Yeah, good morning.
I am a teacher from Griffin International School.
So requesting the.
Yes, sir. Yes, sir. Pardon?
No, please go ahead.
So from
Please
my school, sir, ma'am, Shivam Agrawal.
Which school, sir?
Griffin International School, Kharagpur.
Okay, team D. Kindly, can you just repeat the name, sir?
Shivam Agrawal.
Okay, okay. Shivam Agrawal. Okay.
Okay.
Okay. Okay. The other schools?
Yeah, hello. Good morning to everyone. I am Mrs. Palsara from KV number 2.
Yeah.
My students are Shriyoshi Raha, Nandini, and Ajay Tripathi. So Ajay Tripathi will be the team captain. They are locked in from different places because we are having holidays. So
yes. Okay, fine. Thank you, ma'am.
Hello, good morning, everyone.
Yeah, good morning.
I'm from KV number 1, Kalaikunda.
Yes.
I'm a teacher and I have 3 students. And as my colleague previously said, that they are located at different positions because the school is closed for today.
Yes, ma'am. Yes, ma'am.
I would like to nominate Saksham from my school as the team leader. Thank you so much.
Thank you, ma'am. Aditya Birla Public School, DAV Model School, and Don Bosco.
I'm from AVPS, ma'am. I'm a student, ma'am. From our school, ma'am. Sai Panchanan could be the captain.
Sorry, I didn't get the name.
Ma'am, Sai Panchanan.
Sai Panchanan Biswal, ma'am.
Okay.
Sai Panchanan Biswal, Arjun Mitra, and Mujibur are the team members.
And the captain is Sai Panchanan.
Sai Panchanan, yeah. They all have logged in from 3 different devices, 3 different locations.
We get
Thank you so much.
Thank you. And DAV Model School and Don Bosco?
Yes, good morning, ma'am. From my school, Dipti is there. Dipti Goswami. She will be
which school?
DAV Model School, IIT Kharagpur.
Dipti, okay. Don Bosco? Is there somebody from Don Bosco?
Yes, ma'am, I am.
Yeah, please, who is the captain from your team?
Ma'am, I think that there are, instead of 3 numbers, there are 2 students now. Present, I think that 1 more student could not attend.
Okay, fine. Just give me the name of one person whose answer I can take as final. That's all.
Okay, ma'am. I'm Shaunak. Shaunak Majumdar.
Okay. Thank you. So I think it's good to go, and we'll start with the first round. So in the first round, there will be 5 questions for each team, and the questions will be given at a time. So I'll read out the questions and as well as display it on the screen, and one after the other. So 1 point for each question, and then we move on to the next team. If you have any questions, just get back to me.
Yeah, ma'am, I'm having a question.
Yeah, sure.
Ma'am, is there any negative marking?
No. Yeah, there's no negative marking, and also there is no passing of questions. So the team the question is asked to, that's the end of the question, and we'll give you the answers then. But there isn't any passing of questions, and also there's no negative marking. So you can just take your attempt in the questions. No issue.
Okay, ma'am. Okay.
So I'll just repeat a couple of most important points. Like the team for which the question is being asked for will be the only team which unmutes themselves. Otherwise, it will be extremely difficult to conduct the quiz. And I think, if it will be great if all of you can please turn on your videos so that everybody is on the YouTube channel as well, and here as well, everybody is able to see each other. Thank you.
So if everything is clear, can we just begin? So it's the first round for the team 1, team A, that is Aditya Birla Public School. Are you guys ready? Should I start?
Yes, ma'am.
Yes.
Okay.
Shaunak, you have mentioned the time limit, right?
Sir, the time limit, yes, sir. Just a minute, sir.
Sure. So each question will have a time limit,
yes, sir,
within which you have to answer. Otherwise, you will not be able to answer. Otherwise, the answer will not be even considered as well.
So for the 5 questions, I'll just give you like 30 seconds, or I'll read up the questions, and for each question, take just 5 seconds to answer. Okay, and then we move to the second. So Aditya Birla School students are ready?
Yes, ma'am.
Yeah. So the first question for the team A, the full form of DNA is. So what is the full form of DNA?
B.
B. Can you just spell the answer?
Ma'am, deoxyribonucleic acid. Option number B.
Okay, thank you. That's the correct answer. Second, what replaces thymine from DNA in RNA? What replaces thymine? So thymine is the nitrogenous base which is in DNA. So what is it being replaced in the RNA? Any guesses? I can take your guess.
Ma'am, cytosine.
Is the final answer?
Yes, ma'am.
No, that's unfortunately not the answer. So it's uracil.
Okay.
Okay. So third question, the genotypic ratio of a monohybrid cross is. So what's the genotypic ratio in the monohybrid cross?
Ma'am, it is 1:2:1. A.
Okay. A final answer?
Yes, ma'am.
Yeah, okay. That's the correct answer. Absolutely correct. So now we go to the fourth question. The nitrogenous bases present in the RNA are. So what are the nitrogenous bases present in the RNA? Just take an attempt and take a guess. No issues.
Option A.
Option A?
Yes, ma'am.
So you are saying it's thymine, guanine, adenine, and cytosine, but
yes.
That's the final answer?
Yes, ma'am.
Yeah, but unfortunately, that's not the answer. So answer is uracil, adenine, guanine, and cytosine. So thymine is replaced by uracil, as I already told. So the answer is D. So that's not the correct answer. So now number 5, or the last question. Pea plants were used in Mendel's experiments because. So what's the reason that pea plants were used in Mendel's experiment or not any other plant?
Ma'am, D, all the above.
All of the above.
Final answer?
Yes, ma'am.
Yeah, but it's unfortunately the answer is B. They had contrasting characters. That's the correct answer.
Okay.
Okay, thank you, team A. So now we move on to team B. I think my fellow friends are keeping the scores, right, Mandrita?
Yeah, I'm keeping the scores.
Okay, thank you. So we go to team B. Team B, you are ready? DAV Model School?
Yes, ma'am.
Okay,
yes,
fine.
ma'am.
So your first question is, Autosomes, also known as sex chromosomes. So is this statement true or false?
False. 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 False.
Okay, fine. So that's the final answer?
Yes.
Yes.
Okay, thank you. That's the correct answer. Absolutely correct. Absolutely correct. So the human body cells has how many chromosomes?
46. 46.
46. 40,
Option D,
46.
46.
That's the final answer?
Yes.
Yes,
Okay.
46.
Thank you. That's the correct answer. The third question, the sugars present in DNA and RNA are, so what are the sugars? We talked about nitrogenous bases. So now what are the sugars present in DNA
Option
and
D.
RNA?
Option D.
No, 1 minute. 1 minute.
Ma'am, please give us a minute.
Yeah, for sure.
So
I'll take the answer
we
from
have
Shona.
5 seconds time limit, right?
Yeah.
In case it exceeds, we have to move on to the next question.
Yeah, next question. So,
Yeah,
Shona,
please.
I take the answer
Option D.
from
Option
you.
D. Option D.
Option D is the final answer?
Option C.
Option A.
Option A.
Final.
Option A
think-
final.
I think time is over. I can't
is
give
over.
many
We
more.
move on to
Yeah,
the next
time's
question,
over.
please.
So D is not the answer, which was your final answer. The answer is C. It's a deoxyribose and ribose.
Ma'am, excuse me. Actually, the leader of my school, that is Dipti C, told on the option C. She was going for that. So you
Yes,
should
ma'am.
consider her answer.
But you took more than 5
So,
seconds. I'm
yeah,
sorry.
the time limit was exceeded, so there was a
It was 5
discrepancy.
seconds.
Let's move on to the next question.
Next question. So adenine has the ability to form bond with thymine as well as uracil. So is it true or false? Give me one answer and quick.
True,
Like 5
true.
seconds. It's ticking.
True.
True?
That's true.
Okay, fine. That's correct. So now we go to the next question. And the last question for team B. The phenotype of an organism is, what is meant by
Observable
phenotype?
features.
Option D.
Option D.
Option D. Option D.
Okay.
Observable.
Thank you. That's absolutely correct. Thank you, team B. Now we go to team C. Team C, are you ready?
Yes, ma'am, ready.
Don Bosco, ready? Yeah.
Yes, ma'am.
So this is the first question. In human males, all the chromosomes are perfectly paired except one. This or these unpaired chromosomes is or are. So what is the answer? Is it the large chromosome, the
Ma'am,
small
C.
chromosome? Sorry?
C.
C, 3 and B. So you mean X chromosome and Y chromosome. Is that your final answer?
Yes, ma'am. Yes, ma'am.
Okay, fine. Absolutely
Yes,
correct.
ma'am. Yes,
So
ma'am. Yes,
how
ma'am.
Next question. How many sex chromosomes are present in a human being? What is the number of sex chromosomes in human beings? That's your
Ma'am,
question.
one pair, one pair.
One pair.
What
One pair.
is the option?
Ma'am, A.
Ma'am, one pair, one pair.
What? That's your final answer?
Yes, ma'am.
Yeah, okay. So that's absolutely correct. One pair is the answer. Third question. Which word describes an individual who has two identical alleles for a particular gene?
Homozygous. Homozygous.
Homozygous, homozygous, homozygous.
Give me the
Option
option.
B. Option B.
B.
B,
Final?
B.
Okay.
Yes, ma'am.
That's absolutely correct. Good going. So the I think this is the fourth question. What will be the genotype resulting from? I'm sorry. Yeah. What will be the genotype resulting from a gene cross between two individuals, one of which is homozygous dominant, that is TT, and the other is heterozygous? So what will be the genotype resulting from a gene cross between two individuals?
Ma'am, I think D. Ma'am, option D.
Option D, 25% capital T, capital
Yes,
T, that is
ma'am.
dominant.
Yes, ma'am.
Are you sure?
Yes, ma'am. Yes, ma'am.
Okay, so time is over. I take your answer as D, but unfortunately that's not the answer. It's C. So it's 50% of dominant TT and 50% of heterozygous TT. Next question. In males, the gene for color blindness is located in which chromosome? It is located in which
X chromosome, ma'am, X.
So it is option A, you are saying?
Yes, ma'am.
Okay. I take that as your answer?
Shanak, wait. I think.
Ma'am, it is X chromosome.
Okay.
So it's final. I take Shonak's answer, right?
Yes, ma'am.
Okay. Thank you. That's absolutely the correct answer. Thank you, team C. We move on to team D. Team D, are you ready?
Yeah, ma'am.
Griffiths International School, are you ready?
Yes.
Yeah, ma'am.
So this is the first question. The sex gametes are always diploid, haploid, either haploid or diploid or haplodiploid. So what is the sex gametes are? Any guesses?
Ma'am, please lock option A, diploid.
Option A, lock A, diploid. Fine. But it's not the correct answer. The correct answer is diploid. Not diploid, sorry. The correct answer is haploid. Sex gametes are always haploid. Hence, they come together to become diploid after fertilization. Your next question, guanine bonds with cytosine via, so which is the correct answer? How many hydrogen bonds do you think are single hydrogen covalent bond? Sorry?
Ma'am, single hydrogen bond, option C.
Option C? Sure?
Yeah, ma'am.
Should I take that as a final answer?
Yeah, ma'am.
Okay. So they are saying it's C, single hydrogen bond, but unfortunately that's not the answer. It's 3 hydrogen bond. That's option B. We go to question 3. That is DNA is organized into individual units called. So what are the individual units called in which DNA is organized? Any guesses? So you can take a guess.
Ma'am, we'll go with the option. Ma'am, D.
Option B, RNA.
Ma'am, option D. D for
D chromosomes.
Yeah, ma'am.
Take it as an answer.
No, ma'am. Option A. Option A. Microtubules.
Option A, microtubules. Just give
Yeah,
me
ma'am.
one answer. I'll take it from Shivam.
Option A. Option A.
Option A. Fine. So Shivam is saying option A, but unfortunately that's not the answer. If they had previously told it correct, it was chromosome. No problem. We'll go to the 4th question. A person with the genotype capital A is, what is this type of genotype called? Is it homozygous dominant, homozygous recessive, heterozygous, or haploid? What is the answer?
Haploid.
Ma'am, option A. Ma'am, option A. I take the answer from Shivam, I think. It's
Ma'am, option A.
Ma'am, option A. Option A, homozygous dominant?
Yeah, ma'am.
Okay, fine. So that's the correct answer. Good. Last question for team D. Which of the following cells does not carry any DNA? Red blood cells, white blood cells, embryonic stem cells, and mesenchymal stem cells.
Ma'am, I will go with option B, white blood cells.
White blood cells. I take the answer from Shivam.
Ma'am, just 1 minute.
Yeah, I'll just give you 5 seconds. I don't have 1 minute. I'm sorry.
Yeah, ma'am. We'll go. Option B.
Option B, white blood cells.
Yeah, ma'am.
Okay, I take that as the answer, but this answer is RBCs, or the red blood cells, because they have no nucleus, so no DNA and mitochondria. So that's the answer. I'm sorry, it's the wrong answer. Now we go to team E. Team E, Kenjay Vidal, are you ready? Should I proceed?
Yes, ma'am.
Yes, ma'am. Yeah. So first question is, which of the following type of nucleotides is not present in DNA? Which of the following nucleotide is not present in DNA? Is it adenine, guanine, cytosine, or uracil? Which is not present in DNA?
Ma'am, guanine.
Give me the option.
Ma'am, option B.
Are you sure? Should I take it?
Yes.
No, ma'am, wait.
I have to take Shaksham's answer, I think. He's the captain. So just give me the final answer. Time is up. Yes.
Saksham, take D. It's uracil. Ma'am, yes, ma'am. It's option D.
Okay, fine. So I'm taking answers as D. That's the correct answer. Next, we go to second. Purines are dashed nitrogenous bases, whereas pyrimidines are dashed. So you have to tell me what type of ring structure is present in the nitrogenous bases in purine as well as pyrimidine. So is it single, double, 5, 9, double, single, or single 5? What is the answer? How many nitrogenous bases, like what ring nitrogenous bases are present in purine and pyrimidine? Fast, I think it's time sticking. Give me the answer.
Ma'am, option.
Yeah, I'll just take one.
Option A.
Saksham's answer I have to take. I think he's the captain.
The time is already up. Saksham has to answer, please.
Yeah, and time is up. So I think I'll give you the answer.
Let's move on to the next question, please.
Next question. So this question I'll keep as cancelled. So we go to the next question. Ribose sugar is a what type? How many carbons are present in ribose sugar? 6, 4, 5, 7.
Ma'am, 4 carbon sugar.
Saksham, is your answer B? Give me one answer, I think. Saksham.
Ma'am, 5 carbon sugar. Saksham, take 5.
Sure? I take 5?
Yes, ma'am.
Yeah.
Carbon sugar.
Okay. So that's the correct answer. Now we go to the 4th question. That is, if a plant has round seeds, has a parent that has wrinkled seeds, what is this genotype? Assume that round is dominant. So the plant has round seeds and has a parent that has wrinkled seeds. So what is this genotype? Given that round is dominant.
Capital R and small r.
So you're telling B. Saksham, discuss and give me the answer in 2 seconds. Saksham?
Ma'am, B.
Ma'am, B.
B. Okay, fine. So I take your answer as B, and that is absolutely correct. And this is the last question. DNA from any species of an organism should have a 1:1 stoichiometric ratio of purine and pyrimidine. So this rule is known as, who gave this rule that the DNA from any species should have a stoichiometric ratio of 1:1 for the purine and pyrimidine basis? Which is that? Chargaff's rule, Mendel's first law, second law, or third law? Time sticking.
Second law.
Which law? I think time's going to be up fast, Saksham. Option give me.
A.
Decide among yourself and please tell me.
The time is already up. Now we do not have a final answer from Saksham. So
Saksham. So we pass this question also. So I think now we go to team F. Team F, are you ready?
Yes, ma'am.
Vidyalay 2, are you
Yes,
ready?
ma'am.
Should I proceed? Yeah,
Yes,
okay, fine.
ma'am.
The first question. Somatic cells are all the cells in your body except egg and sperm. So is it true or false? Is it true or false?
True.
True.
Option
A.
A.
True.
Yes.
Okay, thank you. That's the correct answer. DNA can replicate itself. Is it true or false?
True.
Am I having the answers from Ajay? Like, is it
Yes.
a consensus? Okay, okay, fine. So yeah, it's true. Option A is the correct answer. Good. The double helical structure of DNA was discovered by, who discovered the double helical structure of DNA out of all these scientists? Watson, Crick, Mendel, Chargaff, Sanger, and Hargobind Khurana. Who do you think discovered the double helical structure? Time ticking fast.
Ma'am, A.
A. Ajay is also in consensus
Yes.
with A.
Yes, I'm also going with it.
Okay, okay. Thank you. That's the correct answer. The central dogma of molecular biology is, so what is the central dogma supposed to mean in molecular biology? RNA makes DNA, DNA makes protein, protein makes RNA, DNA makes protein, or DNA makes RNA, RNA makes protein, or DNA and RNA both can make protein. So what's your answer for the central dogma of molecular biology?
I'm C.
C? Sure?
Yes.
You take a correct answer? So yeah, that's absolutely correct answer. DNA makes RNA and RNA makes the protein. That's what central dogma means. So your last question, an organism with alternate forms of a gene will express the form that is dominant. What is this law known as? So what is this law known as? An organism with the alternate forms of a gene will only express the form that is dominant. Which law means this? The law of segregation, law of independent assortment, law of dominance, or law of inheritance?
Law of dominance.
Are you sure? Should I take that as your answer?
Yes, ma'am.
Yeah, so time's up, but now that's not the answer. The answer is the law of segregation. So I think that's all. Thank you guys for that. And I'll ask Mandrita to share the course and we'll just proceed to the second.
thank you so much, Shadrupa. That was a very well conducted session.
So Mandrita, can you just give this course, and then if there's a tie, we can go for the tiebreaker if we need it.
Yeah, so we don't have a tiebreaker situation.
Okay.
So I'll just announce the scores. Aditya Birla scored 2 points.
Okay.
DAV scored 4 points. Don Bosco scored 4. Griffin International 1. KV 1 3. KV 2 5. So in this round, Griffin International gets eliminated.
Okay. So we now proceed to the second. I'll just stop sharing the screen.
Sorry, can you please announce who got eliminated in this round? One more time.
Team D. Team D. Yes, Griffin International School got eliminated. So we proceed to the second round.
Yeah, I mean, it's unfortunate, but well played, Griffins International. So from here on, we will have 5 teams, right?
Yes, sir. So we'll take 5 teams forward to the second. And now I'll ask Harshit and Mithun to take up from here.
So requesting all Griffins students to please be here and be a part of the session and listen to the questions. Well played. And yeah, let's move on to the next one. Thank you.
Yeah, good morning to one and all present here. So I'll be conducting the next round. I'll share my screen. Okay. So the next round will be a Pictionary round where you'll be given images and okay. Yeah. So the next round will be an image round. So where you'll be given an image and you have to identify the image. So coming to the rules. So there will be one question per team, and each question, each correct answer will carry 1 point, and the questions will not be passed on, and there will be no negative markings. And the time limit for each question is 10 seconds.
So here I would just request Mithun, please maintain the time limit.
Yes.
And also to all the participants, the final, whatever is being said by the quiz master is the final verdict here. So we would not request you to like question after that. And the time limit will be maintained, please, very strictly. Okay. And the answer from the captain will be taken as the final one.
Yes, sir. So I would like to make an announcement regarding the team names because one team got eliminated in the previous round. So team A is Aditya Birla School, team B is DAV, team C is Don Bosco, team D will now be KV1, and team E is KV2.
Thank you.
So I'll be moving on with the questions. So team A, Aditya Birla. So the captain, you can choose a number. So each number will be leading you to a question. So you can choose a number.
5.
5. Okay. So identify these set of laws. What are these laws called as?
The time starts now.
Mendel law of inheritance. Mendel law of inheritance.
Okay. Yeah. So that's the correct answer. So moving on to the next team. Team B, DAV Public Model School, KGP. The captain, Deepthi, can you please choose a number?
Sir, second question.
Okay. So what is a single helical structure called as?
RNA.
Yeah.
Right.
Okay. That's the correct answer.
Okay.
So moving on to the next team. Team number C, Don Bosco KGP. Shaunak?
So 1. So number 1.
Okay. So what are these structures called as?
So the structures are called as chromosomes and the full arrangement is called as karyotype.
Okay, you're right. So moving on to the next team. Team D, Kendriya Vidyalaya 1, Kalaikunda. Shaksham, can you choose a number?
Yes, sir.
Yeah.
And number 4.
Okay. So what is this double helical structure called as?
Deoxyribonucleic acid DNA.
Okay, that's exactly right. So moving on to the final team. So team number E, Kendriya Vidyalaya 2. Ajay, can you choose a number? 3. Okay. So what is the square diagram to study breeding called as? What is the name of this square diagram? You have 10 seconds.
Punnett square.
Yeah, Punnett square is absolutely right. So all the teams have got the questions right in this round. So moving on to the next round, I'll be handing it over to my colleague, Dr. Harshita. Thank you.
Well played, all the teams.
Thank you.
Is my screen visible?
Yes.
Yes.
Okay. So moving on to the next round. This is a murder mystery round. So this is going to be interesting. And it's called as Let's Get Sherlock. So I'll be briefing about the rules once more. So yeah, after we go through the story, which is our murder mystery, each team will get one question and 1 point for each correct answer. There will not be any negative marking and there won't be any passing of questions. And time limit for each question is 10 seconds. So this is our story. So I will be reading it once and then I'll give you like 10 seconds after which you can go through it once more. And then, yeah, we'll be going on to the questions. So a man named John walks into his house. It was pitch dark, but something seemed off. There hasn't been a day since Tom, his retriever, welcomed him the moment he entered the house every day. He tried switching on the lights, but it was odd that there was a power cut. Strange that such a thing occurred after many years. He somehow managed to fumble for a torch that he always kept handy. He switched it on and was astonished to find blood on the kitchen floor. There was no doubt that there were signs of struggle since all the cutleries and bowls were on the floor. But what made him scream was the dead, mutilated body of a man that was lying on the floor. The darkness prevented him to recognize the person, but even the torchlight wasn't quite helpful. The face was covered in blood and the murder weapon was still lodged in his chest. But who would possibly kill some other person in his house? Nevertheless, he managed to call the police, shaken to his core. Tom was missing too. The body was taken for further investigation and autopsy. Samples were taken from hair, blood, and oral mucosa. Meanwhile, John called his mother over to support him through this traumatic incident he faced. After some days, some interesting clues came from the forensic department. So the story will be displayed for another 5 to 10 seconds, after which we'll move on to the questions. Okay, so time's up. So we'll be moving on to the question. So each team will have to choose a nucleobase, and based on that, the questions will be displayed. So first, team A, Aditya Birla,
Yes, ma'am.
would you like to choose a nucleobase?
Ma'am, guanine.
Okay. This is your question. It was revealed by the forensic experts that the murder could not have been successful if it were not for the profuse, uncontrolled bleeding of the victim. Later, while interviewing John and his mother by the detective, the mother confessed that she was a carrier of this particular disease, which John and his twin both had. So what was the disease? These are your
Option
options.
A, hemophilia.
Okay, let's see the answer. Is that your final answer?
Yes, ma'am.
Yes, ma'am.
Okay, you got it right. Good job. So, coming back. Team B, DAV Model School.
Yes, ma'am. Ma'am, I would like to select guanine.
Guanine is already done.
Oh, okay, ma'am. Thymine.
Okay. So this is your question. As John was the first person in with the police, took his fingerprints and saliva. To his surprise, the person murdered had a match with John's DNA sample up to 99%. So who really was the person murdered? Is it his brother, fraternal twin, identical twin, or father? Your time starts now.
Father.
Um,
Father, I think. Father. It might be father.
Ma'am, option D.
Give me one answer, please.
Option D.
Option D. Father.
Option D,
Father.
father.
Option D, father.
Okay, let's see the answer. I'm sorry, the answer is identical twin. Okay, moving on. So, Team C, Don Bosco.
Ma'am, I want to choose adenine.
Okay. So, this is your question. What was extracted and analyzed from the samples of the dead body? And your options are protein, RNA, DNA, and microRNA.
DNA.
Is that your final answer?
Yes, ma'am.
Yes, ma'am.
Okay. And you're right. Moving on. Team D, KV1. So the remaining ones are cytosine and uracil. You can choose one of this. KV1, are you there?
Ma'am, cytosine.
Okay. So this is your question. Some days passed as the investigation took off. The blood samples gave some confusing results upon further analysis. The DNA sample from the blood acquired from the knife showed a DNA similarity to humans of around 80%. Who could the other victim be? So these are your options: John's second cousin, Tom, who's John's retriever, John's neighbor, or John's father. And your time starts now.
John's second cousin, option A.
Okay, is that your final answer? Okay, your time's up. Let's see the answer. It's Tom, John's retriever. Okay, moving on. So KV2, I think you're left with Uracil. So yeah, this is your question. So with the twist of events, the murder weapon analysis gave surprising results. The DNA from the fingerprints on the weapon had considerable similarity with John's DNA, up to 12%. So who is the killer? Is it the mother, father, brother or sister, or cousin? And your time starts now. KV2.
I'm thinking cousin.
Okay, I take that as your answer since time is up. Yes, and you're correct. So this comes to the end of murder mystery round. So I'll be handing over to Dr. Mithu.
Wonderful, Dr. Harshita. Very well conducted. So can we have the scores after this round?
Yes,
Yes,
sir.
just a second.
Sure.
Okay, so Aditya Birla has 2 points, DAV 1, Don Bosco 2, KV1 1 point, and KV2 2 points.
I just wanted, like, the cumulative scores are also being calculated, right?
So that's the cumulative score of the last 2 rounds.
Okay, okay, fine. Thank you.
Yeah.
So we'll be moving on to the next round, which is called as Who Am I? So going to the rules of the round. So as the previous rounds, there will be one question per team, and each correct answer will carry 1 point. And the questions will not be paused on, and there will be no negative markings. And the time limit is the same as before, 10 seconds per question. So going to the questions. So each team has to choose a cell organelle. So based on that, it will lead to a question. So let's start with team A. Aditya
Sir,
Birla.
choose mitochondria.
Okay.
Mitochondria.
So this is your question. So DNA to DNA is replication. So what will be DNA to RNA?
Transcription. Transcription.
Transcription. Can I take that as a final answer?
Yes, sir.
Okay. So the correct answer is transcription. Yeah, you're right. So moving on to the next question. DAV model school. Can you choose an organelle?
Yes, sir, Golgi body.
Okay, so this is your question. So DNA to RNA is in the nucleus. So RNA to protein, where will it be? You have to guess this.
It might be endoplasmic reticulum.
Okay.
Endoplasmic reticulum.
Sir, yes, sir, endoplasmic reticulum.
Endoplasmic reticulum.
Endoplasmic reticulum. Rough endoplasmic reticulum.
Okay, so unfortunately, that's not what I was looking for. It is a cytoplasm, or if you had to be more specific, it could have been the ribosome.
Sir, but ribosomes are present in rough endoplasmic reticulum only.
Okay.
No,
Okay,
so
yeah.
the quiz master's answer will be taken as the final one, and yeah, so ribosome is the most specific answer, and here we are not going to discuss about the answers. Please go ahead.
Okay, but I think we can take rough endoplasmic reticulum as also the answer, since it also contains ribosomes. Okay, so team C, Don Bosco KGP.
Sir, nucleus.
Nucleus.
So DNA is to RNA, RNA is to what?
Shanak, I think protein.
Protein. Protein.
Okay, so let's look at the answer. The answer is protein. So moving on to the next team. Team D, Kendriya Vidyalaya 1. Can you pick an organelle?
Lysosomes.
So this is your question. So DNA is to ATGC, RNA is to what? You have 10 seconds. Hello?
Mithun, if the time is up, we move to the next person.
Yes.
Yeah. No answer.
Okay, so since we didn't have an answer, we'll look at the answer. So these are basically the nucleotides, nucleotide bases present in the present in RNA, which are AUGC. So moving on to the final team, which will be Kendriya Vidyalaya 2. Can you pick an organelle?
What all is left?
You have endoplasmic reticulum and the ribosome.
Endoplasmic reticulum.
Okay. So this is your question. DNA is to double-stranded. What is RNA?
Single-stranded.
Okay, let's look at the answer. So you're absolutely right. So that's the end of this round.
Wonderful round, Dr. Mithun.
Thank you.
So, Mandita, if you can update us with the scores.
Yes, sir. So the cumulative scores for round 2 is Aditya Birla has 3 points, DAV has 2, Don Bosco has 3, KV1 has 1, and KV2 has 3. So in this round, DAV model and KV1 are getting eliminated. And the Aditya Birla and Don Bosco and KV2 are moving on to the final round.
Okay. Unfortunate for the ones who are getting eliminated after a well-fought battle so far. But as per the format, we have to move ahead. So congratulations to the ones who have qualified for the next round.
Yeah, so we start with the final round with 3 teams. So that will again, I'll just share the screen. So the first, this round 3 is also going to have 3 sets of questions. So first we'll be doing experimental set of questions. So here the rules are again same, the 1 point for each answer. There isn't any passing of questions, and I take the answer from the captain as your final answers. So the first team, so who is going to be the first? I'll just, Mandita, can you just update?
Yeah, I'm updating. So team A becomes Aditya Birla, and team B is Don Bosco, and team C is KV2.
Okay, fine. So we begin with Aditya Birla then. Aditya Birla, are you ready?
Yes.
Okay, so this is your question. You cross a plant having red flowers with white ones. You see the next generation gives flowers that are red in color. What trait do the red ones have?
A, option A, dominant.
A, dominant trait.
So is your final answer A, dominant?
Yes, ma'am.
Yes, ma'am.
Absolutely correct. So we go to team B now, that is Don Bosco. You isolate some tissues from a rat's tail and its ears and some from its liver. Then you extract the DNA present in those tissue samples and analyze it. So the DNA from these entire 3 tissue sources will have different genetic code. The following sentence is true or false. So you take the DNA from 3 different things, different parts. So is it going to have the same genetic constitution or different? Is it true or false?
True.
Your answer is A, true.
I think.
Yes, ma'am, true.
Yes, ma'am, true.
Is your final answer? Okay, fine. I take your answer is true. But unfortunately, that's not the correct answer. It's false because once you take the rat's DNA and human DNA cannot be same. So Next, we go to team C, that is KV2. You have another question related to similar type. You isolate some tissues from a mouse and a rat's tail. Then you extract the DNA present in those tissue sample and analyze it. So the DNA from these two entire tissue source will have the same genetic code. Is it true or false? So you take it from a mouse and a rat's tail and analyze the DNA. So are they having the same genetic constitution or not? Is the following statement true or false? I think time's up.
False.
False.
Okay. Should I take the answer?
Please.
I leave that to the quiz master.
Okay, fine. It is like 1 or 2 seconds. Fine. Okay, the final answer is false. So that's absolutely the correct answer. So we are done with the experimental part, and I'll pass on to Mithun or Harshita to take up the next round.
So how many rounds are we left with?
Two more, sir. Just 2 more.
Okay, excellent.
Yeah.
Great going.
Harshita,
Okay.
Mithun, please take up.
Yeah. Okay. So I hope the screen is visible.
So after this round, we should announce the scores and then we move on to the final round. So to make it more interesting.
This is the first part of the final round only, which we just conducted.
Okay, no. So can we then have the scores so that
Yeah,
all
sure.
the teams have an idea, because it's like converging
Mandita,
towards a close finish.
Yeah, Mandita, give the cumulative score of all the 3 rounds, like beginning from the beginning.
Okay. So 1 second. Just give me 1 minute.
Yeah, sure.
Yeah, please take your time. So do we have like 2 rounds in this round, within this round, or how is it going to be?
No, within this round, 2 sub rounds. So that's just 2 set of different questions. Yeah.
So in that case, we can move with the first sub round until which Mandita can be calculated.
Yes. Harshita, please take up. Let Mandita calculate till then.
Yes.
Okay, so this is going to be more of a history-based round, like who discovered and who invented, etc. So it's called Blast from the Past. So these are your rules again. Each team will get 2 questions, 1 point for each correct answer. There's no negative marking and no passing of questions. And the time limit is again 10 seconds per question. So each team will get to choose 2 amino acids, and based on what they choose, the questions will appear. So we'll start with team A, Aditya Birla.
Which one you will choose?
Valin.
Sorry,
Number 5.
which one, please? Can you repeat?
Number 5, Valin.
Number 5, Valin. Okay. So you're choosing Valin. Okay. So this is your question. With whom you can associate the theory of natural selection? And your options are Barbara McClintock, James Watson Francis Crick, Gregor Mendel, and Charles Darwin.
Option D.
Charles Darwin.
Charles Darwin. Yes, ma'am.
Okay,
Option
let's
D.
see the answer. And you're right. Okay, so okay, next is team B. Team B, could you choose an amino acid? Valin is done.
Glycine, first one.
Glycine. Okay. So this is your question. Who is known as father of genetics? And your options are
Gregor
William
Mendel.
Johnson.
Gregor
Okay.
Mendel. C.
Okay, let's see the answer. You're right. So moving on. Team C, KV2.
Alanine, number 6.
Alanine, okay. So this is your question. What was the first cloned animal from an adult cell? Dolly the sheep, blowfish, horse, or liger? And your time starts
Dolly the
now.
sheep.
Is that the final answer?
Yes, that is the final answer.
Okay. And you're right. So all the 3 teams have gotten the correct answer in round 1 of this round. So moving on to the next. So again, team A will have to choose another amino acid. Valin is done, glycine is done, and alanine is done.
Number 4.
Number 4. Okay. So who won the Nobel Prize in Physiology or Medicine for discovering the genetic code? These are your options.
Ma'am, option A. Ma'am, option C.
Okay, is that your final answer?
Yes,
Yes,
ma'am.
ma'am.
Option C.
Yes, ma'am, option C.
C. Okay.
And D.
Unfortunately, it's option D. I'm sorry, I won't be taking that answer. Okay, moving on. Team number B, sorry, team B, Don Bosco.
And the third option is done. Yes, reading that one.
No, that's not done.
Ma'am, say which was third one.
Okay. So who coined the term mutation? Francis Crick, Hugo de Vries, Erwin Chergaff, or Charles Darwin? Five seconds are up.
Ma'am,
Ma'am, I
B, ma'am,
think
B, ma'am,
it
B,
is
ma'am, B.
Yes, ma'am. Yes, ma'am. B.
Ma'am, B. B.
Okay, fine. It's the right answer. So team C, you have tyrosin remaining, so I guess that will be your amino acid. So who discovered jumping genes? Francis Crick, Rudolf Janisch, John Baptist Lamarck, and Barbara McClintock. Your time starts now.
Barbara McClintock, D.
Yes, that's the right answer. So that comes to the end of this round. So next we'll be having rapid fire. Dr. Mithun will be briefing about the rules of the rapid fire round.
Thank you so much, Dr. Harshita. Let's just wait for a while if Mandrita has the scores until the previous round.
Yes. Okay, so previous round the scores were something like this. Aditya Birla scored 6 points, Don Bosco scored 7, and KV2 9. So you are not audible.
Sorry, I was mute. So this was the score that was prior to the round that was conducted by Dr. Harshita.
Yes.
Okay, and so I believe all the teams now are almost aware of where they stand. Good luck with the final round. You have already gone through the first phase of this final round, and then the next phase is the one that is remaining. So all the very best to all the three teams. Good luck, and please go ahead now.
Okay, so for the final round, it's the rapid fire round. So I'll be briefing the rules to the teams. So there will be 10 questions per team, for which the time limit, sorry, not presenting. Okay. So the rules are as follows. There will be 10 questions per team, for which the time limit will be 1 minute. The total time limit will be 1 minute. So for each correct answer, there will be 0.5 points, and there will be no negative markings. So you have to move through the 10 questions within the stipulated time of 1 minute. And if you don't know the answer to a question, you can say pass and move on to the next question, so that you don't waste your time on a single question. Yeah. Okay, so yeah, these are the types of RNA. So you can choose any one type, and based on that, you will get a set of 10 questions. So team A, Aditya Birla, you can choose. Yeah, hello?
mRNA.
mRNA. Okay, so I'll hand it over to Harshita to start with the questions.
Okay, so the questions won't be displayed since it's rapid fire. I will be reading out the question, and you'll. It's about 1 minute, and there'll be 10 questions. So the faster you answer, the more questions you'll get to. I'll read out. So and the points will also increase accordingly. So you've chosen mRNA, right?
Yes, ma'am.
Yes, ma'am.
Okay. So let me know when you're ready. Then I'll start the timer and start reading out the questions. Are
Yes,
you ready?
ma'am. Yes, ma'am.
Yes, ma'am.
Okay, so I'm starting the timer for 1 minute. It starts now. Who discovered particulate inheritance theory? Be quick so we can move on to the next question. If you don't know, just say pass.
Pass.
How many pairs of characters of garden pea plant did Gregor Mendel choose for his experiments?
7.
Okay, that's correct. Next question. What charge does DNA carry?
Can you repeat once, ma'am?
What charge does DNA carry?
Positive.
Positive.
That's wrong. It's negative charge. Next question. Two strands are held together in DNA by which type of chemical bonds?
Hydrogen bond.
Hydrogen
Yeah,
bonds.
that's correct. Next question. Which type of inheritance is seen in the disease hemophilia? Autosomal or sex-linked?
Ma'am,
Sex.
Give me one answer, please.
ma'am,
Sex.
Should I take sex-linked as the final answer?
yes, ma'am. Yes, ma'am.
Okay, that's the right answer. Next question. Okay, so I'm sorry, but your time is up. So how many, just give me a second. So you've answered 3 correctly. So so for the rapid fire, you get 1.5 points extra. So moving on to the next team, team B, Don Bosco.
Ma'am, I want to do the second one.
TRA?
Yes, ma'am.
Okay. So let me know when you're ready, then I'll start the time.
Yes, ma'am, I'm ready.
Yes,
Ready?
ma'am.
Okay, so I'm starting the timer. How many chromosomes are present in human gametes? Human gametes.
Ma'am, 23, ma'am, 23.
Okay, you're right. Next question. Sickle cell anemia is a sex-linked or autosomal disease.
Autosomal.
Autosomal.
Okay, you're right. Next question. During his experiments, which term Mendel gave to the genes?
Ma'am, fear.
Factor.
Yes, yes, okay, correct. Next question. How many strands are there in DNA?
Ma'am, 2.
Okay,
2.
correct. How
Double
many
stranded.
types of, okay, next question, correct. How many types of gametes produced by a heterozygous individual?
2.
Okay, correct. Next question. Who was the first test tube baby in India?
Ma'am, pass.
Ma'am, pass.
Okay. Who devised the binomial classification system?
Ma'am, pass.
Okay, and your time is up. So I think you've gotten 5 questions correct. So you get 2.5 points for this round. Good job. So team C, you're left with RRNA. So are you guys ready? KB2?
Yes.
Okay, so I'm starting the timer. How many autosomes are present in humans? If you don't know, just say pass so we can go on to the next question.
44.
44.
Okay, yeah, that's correct. Which plant did Mendel choose for his experiments?
Pea plant.
Okay, correct. Who invented the central dogma of molecular biology?
Pass.
What is the genetic similarity between humans and chimpanzees? I want the percentage. Next question.
90%. Wait.
Okay, I'm sorry, that's wrong. It's 96. Alternate forms of gene is called?
Pass.
Who was the first test tube baby in the world?
Pass.
Okay, your time is up, Team C. And you've answered 2 questions correctly. So you get 1 point for this round. So we'll be displaying the final score in a while.
Wonderful, very well conducted session. Thank you so much to all the quiz masters. Amazing. And the teams have performed incredibly well. So congratulations to all the teams. What a wonderful session this was. I mean, we are still waiting for the final tabulations. So good luck. Let's see how everyone has fared. And I can see the tense faces as well for each of the students. Still waiting. I can understand that this wait is always seems to be the most longest duration when you get to see the final results. So yeah, just a few more moments before we announce the winner.
So they are biting their nails.
Yeah, it does. We can see that. So if the quiz masters would like to take some more time, we can also give that to them. I would just then proceed with the vote of thanks. And once I finish the vote of thanks, maybe we can announce the winners. So the quiz masters, you can please take your time.
Yes, sir.
Let me just proceed with the vote of thanks. So definitely, I mean, what we saw today with the quiz competition, of course, as per the format of such events, there will be some who will be winning, other teams will be losing. But all I can say is at the end, it does seem to me the victory of science. And it is extremely wonderful and very heartening to see all the students, such young students, being so like motivated and interested in molecular biology, genetics. As we have seen over the last nearly 2 years now, what the potential of genetics is in terms of the disease that suddenly came into existence out of nowhere. Nobody had any idea about it. And then the rapid progress in which the vaccines were discovered and the logistical challenges associated with how the vaccines will be given to the different like this population which is close to 7 billion in the world right now. But we have already achieved this remarkable feat to a large extent. Of course, a lot of work still needs to be done. What we have definitely probably have been able to do quite a bit, which wasn't probably kind of expected in yesteryears, the feat that we have achieved right now. There's still a long way to go, but the whole purpose of such an event is to motivate young students and young children to be more interested in the field of biology in general, and more particularly in the field of genetics, because now with the world opening up to science which can do genetic engineering, which is basically engineering your own body's machinery, then definitely this is only a matter of time. There are challenges associated with this, but over time, I'm sure we will be able to progress to a situation where we can probably correct many of the defective genes and the diseases associated with them. So this is just the beginning for you guys. Some of you will turn out to become clinicians. Somebody would be interested to go ahead with a career path in research, academia. And we would love to see that happening out of students from the ones who joined this today's event. But at the same time, we also do understand that there were a lot of challenges when we started thinking and hosting this event in an online platform. And with the online platform, it is not always possible for everyone to be so inclusive. So we did miss out on quite a few schools which we would have loved them to participate, but definitely with all the students living in different parts of the like city, Kharagpur, and again around in across India as well, we would have loved to go throughout the country as well, but there are a lot of challenges associated with the connectivity and all. So we did encounter situations like these, and we approached a lot of schools. They also expressed their willingness, but at the same time, they also express their helplessness in how do we reach out and how do they ensure that the students will be able to connect. So we do understand and we empathize with all of these. Nevertheless, probably what I can say is this possibly is one of the good beginnings that we have had, and it was very heartening to see the motivated students, the teachers who were so helpful from the very beginning in coordinating the entire event. So thank you so much. And of course, thanks a lot to our experts, Professor Praful Chandra Shukla and Dr. Rashmi Sinha, for sharing their valuable time and their expertise in this field to help us understand and make the students much more attracted towards this. I mean, probably at the moment to an extent fictional world, which is slowly moving towards reality. So it means so much for us, Professor Shukla and Dr. Sinha. Thank you for your time and effort. And thanks to all the schools. Especially, again, I would like to mention the names. Aditya Birla Public School, Bhubaneswar, DAV Model School, Kharagpur, Don Bosco School, Kharagpur, Griffins International School, Kharagpur, Kendriya Vidyalaya 1, Kalaikunda, and Kendriya Vidyalaya 2, Kalaikunda. Thanks to the coordinating teachers and their heads, of course, to the students, and especially to the parents who have been holding on to. I'm sure there are so many different kinds of events and activities going on in the schools. So holding on to all the things, all the challenges with so much enthusiasm for all the events, and again, so much enthusiasm for our program. It is indeed very heartening. Thank you so much. We understand how much challenging this online education has been for all of us, and yet we have managed to tide and overcome these challenges for today's event as well. I'd also like to thank all the volunteers for the program, who are our lab members as well at School of Medical Science and Technology at IIT Kharagpur. So they have been working tirelessly towards the execution of this program, and fortunately, it has turned out to be extremely well. So I would especially like to mention a few of the names: Mr. Prasanna Kumar Bairam, Mr. Motiur Rahman, Ms. Lopamudra Das, Ms. Mandrita Mukherjee, Ms. Shatarupa Bhattacharya, Dr. Harshita, Dr. Mithun, Ms. Pravinya Suri, Mrs. Adari Ghosh, Mr. Shukhen Mahata. So thank you so much. Without your time and effort, the amount of work that you have done over the past, I think, close to a month now in putting everything together, thank you so much. Without your contribution, we would never have been able to achieve what we have been able to do today. And of course, a very special thanks to the Royal Society of Biology UK for funding the event. So initially, as I mentioned, the entire event was supposed to happen in an in-person mode. Unfortunately, with the COVID-19 pandemic hitting us so hard, we were not able to, we were overwhelmed, and we could not even think about doing it in an in-person event last year. And then in the early part of this year, we started thinking about it, but then India got hit with the second wave, and so things got stranded. But definitely, thanks to the Royal Society of Biology, they also understand the challenges ongoing, and they were very happy to help us move towards an online platform. Special thanks to Ms. Amanda Hardy from the Royal Society of Biology, who has been extremely helpful during the entire grant application process. And she was always so responsive and so prompt in answering to any query that I had, any concerns. She would be very empathetic with the entire situation. So thanks a lot to her. I'd also like to extend my thanks to my INYAS colleagues, and in particular from the Kolkata Bhubaneswar chapter, Dr. Chirashri, Dr. Shriparna, and Dr. Upasna. Thanks to Madhavi from our INYAS office for keeping us together during the entire Zoom session today. And thank you so much. And of course, everyone else who has been a part of it. I am myself, Dr. Nishant Chakravarti. Presently, I'm a member of INYAS, and in particular for the INYAS Kolkata Bhubaneswar chapter, I'm a co-coordinator as well. Thanks to the continuing education program, IIT Kharagpur, for guiding us through each and every step that we took. We sincerely hope everyone enjoyed the event thoroughly. I believed there'll be many more students who might have been keen on participating. And in fact, when we started the event today morning, I started receiving some WhatsApp messages from different schools. I don't know which schools are these as well, who the students were like kind of very much interested to join the quiz competition. Unfortunately, with the way the entire thing was organized, it was not possible for us to accommodate these students at the last minute. So I shared the YouTube link with them, which link will always be there. So I'm really sorry that we couldn't accommodate those students. This shouldn't dishearten any of the students. And this link that we shared with all of you will be available as a permanent YouTube link as well. And we sincerely hope that this is just the beginning and we are able to move on to something bigger and better next time. So thanks once again to everyone. Before we close the session, of course, as we could have seen that all the students were nail biting. So now we would like and we would request Mandrita and the other volunteers to please come up with the final winners list.
Please share your screen. Yeah, so in the third position, it's Aditya Birla Public School with 8.5 points. Second is Don Bosco School with 11.5 points. And Kendriya Vidyalaya 2, Kalaikunda with 12 points. I want to have a special mention to Don Bosco, who played really well in the rapid fire. Unfortunately, 0.5 difference.
Yeah, it's just a very small difference.
So really played well.
But excellent, excellent, amazing. A big round of applause, everyone, for all the teams.
Congratulations to everybody.
Congratulations to Kendriya Vidyalaya, to Kalaikunda for playing so well. And of course, for Don Bosco School, Kharagpur, and Aditya Birla Public School, Bhubaneswar, they also played really well. Unfortunately, as I mentioned, as per the format, there will be a winner and there will be some teams who couldn't reach that, and so maybe just by a fraction as well, as we can see. Nevertheless, the other teams who were not in the top 3 also played very well. Unfortunately, they were not in the top 3, but well done indeed. Thank you so much. Thanks a lot. But before we close the session, I would request everyone to please turn on their videos. You can, Mandrita or anyone who is sharing the screen may please close the screen share so that all the videos are in one place, and then requesting someone to please take a picture. So if you can just place all the, Professor Shukla, if you are here as well, that would be wonderful to have you on our screen.
Yes, I'm there.
Okay.
I wish to, but my setting is not allowing me to start the video, so I am unable to show. But I am here.
That's okay. That's okay. Just we will just try to change the format so that we can take a picture. Is somebody getting it in the grid format? Because even I am not able. Just change the view to the gallery view. Yeah.
I'm taking, sir. I'm taking screenshot.
Okay. Excellent. If you, I think there are 2 pages, so you need to move on to both the pages, please, huh?
Yeah, it's
okay. Thank you so much. Thanks a lot. Thank you very much, and hope we meet again sometime later. And maybe this time it will be an in-person meeting. So, Madhavi, you may please stop the